Well, hello everybody. Welcome back to Monday. Monday. This is day 7 of 11 days of biochemistry, and we're right where I want to be. So I see that some of you in Detroit there, in Columbus, it's raining up there. Rainy days and Mondays always get me down, but today you're not going to get down because we got a lot of high yield stuff coming your way. You're going to be glad that it's raining up there because you have nowhere else to go but to be indoors and to listen to biochemistry. So anyway, we've got a lot of good stuff coming your way, a lot of high yield stuff. You're going to be thankful that you came tonight because a lot of integration stuff with biochemistry with endocrinology, and you're going to see a lot of pathology as well tonight, especially later on in the evening. Okay, so where we're at is, so last week we spent a lot of time talking about genetics and molecular biology, talking about chromosomes and genes and RNA and this and that and all that kind of stuff. But what's that got to do with anything? And what's that got to do with life? And the idea, as we talked about on the last day on Saturday there, we talked about the idea is that in your body, you are a bag of chemicals. Even though you don't like to think about it, but you are a bag of chemicals. And in these chemicals, you have pathways to convert chemicals from making glucose and burning glucose and making fatty acids and burning fatty acids all in the same cell. And all these chemicals in one cell can go into different kinds of other chemicals. So the idea is in these metabolic pathways is but the rates of these chemical reactions are too slow. That's why you have enzymes encoded by genes in 46 chromosomes that make these reactions go faster here. And then, so we talked about the concept on Saturday of why you have enzymes in the first place. So again, to make the reactions go faster. And we talked about the concept that when when one enzyme is missing, then you got a major problem called pathology, because the rate is too slow of a particular reaction. And then if the rate is too slow, the substrate accumulates, and when the substrate accumulates, it spills into other metabolic pathways, causing havoc to the cell, and then the organ, and then the body suffers as a consequence here. So again, the concept of why we have enzymes in the first place. Now, chapter 9, that's where we're at right now, is the issue of why do we have hormones? And the idea, this is major integration with endocrinology. And the idea is, of the hormones that we have, there are two major classes, and one is the fat-soluble hormones or the lipid-soluble hormones versus the water-soluble hormones. Now, the lipid-soluble hormones were already discussed. That was way back in molecular biology. All lipid hormones, they work at the gene level, whether it's cortisol, whether it's testosterone, estrogen, or whatever, they all work at the gene level. And as a result, because they work at the gene level, they work very slowly. Now, in this chapter, we're going to talk about how do the water-soluble hormones work. And because they're water-soluble by definition, water-soluble hormones, what that means is they cannot enter the cell because they're water soluble. They cannot enter the cell. They cannot go through the plasma membrane to come into a cell to turn on a particular pathway. So as a result, the hormone must bind to a receptor on the cell surface of a particular target cell, and then it signals. And then there's signal transduction that ultimately results in the turning on or turning off of a particular pathway. So that's what's going to be discussed in chapter 9, are 3 kinds of signal transduction pathways. Now, the first of the 3 is the signal transduction pathway that's mediated by glucagon and epinephrine. How does glucagon or epinephrine, how do they turn on and turn off pathways? The second of the 3 signal transduction pathways we're going to review in this chapter is how does nitric oxide as well as ANP, how do they signal vasodilation. You know nitric oxide causes vasodilation of smooth muscle, but how does it happen? How does it occur? It's all coming up in the second of the three signal transduction pathways. And then thirdly, we're going to talk about the signal transduction pathway that's mediated by insulin as well as growth factors. How does insulin or growth factor, when it binds to its point of view? Dr. Wilson, several weeks ago, and I guess it was in March when he started the course off, he talked about the physiology point of view of these hormones, but now the biochemistry point of view. So the first one is, again, you got to know this information of how these hormones work. So the first of the three signal transduction pathways is, how does glucagon, how does epinephrine, how do they mediate signal transduction? Okay, so now suppose we have a plasma membrane of a target cell for glucagon or epinephrine. Now, because glucagon is a polypeptide hormone, because epinephrine is a small water-soluble hormone, they cannot enter the cell to turn on a particular pathway. So as a result, these hormones must bind to a class receptor on the plasma membrane of the target cell. Now let me ask you this. What is the target class receptor? What's the target class receptor? Remember, lipid-soluble hormones. Let me ask you this. What do lipid-soluble hormones use? What class receptor do lipid-soluble hormones use? Let me see your answer. What do lipid-soluble hormones use? Not GRE. That's the GRE is the target DNA sequence. But that's right. It's the zinc finger class of receptor. The zinc finger class of receptor, which is also classified as a specific transcription factor class. So again, they are specific transcription factors, but the class of receptors are called the zinc finger receptors. And then when they are turned on by the hormone, they go into the nucleus looking for a piece of DNA called the HRE, also called the GRE, those things we talked about earlier here. So those are response elements, but they are DNA. They're not the receptor. The receptor is the zinc finger class of receptor here. Okay, now the urease, not CRE, not PPREs. Again, that is that's the target DNA. That's not the class of receptor here. Okay, now with these water-soluble hormones, what's the class of receptor here? With glucagon and epinephrine, what's the class of receptor? The class of receptor called? Let me see your answers here. What's the class of receptor? No. Let me say, there we go. And they're called the adrenergic class of receptor. Adrenergic class of receptors. Now, when you the epinephrine binds to the beta adrenergic receptor or the alpha adrenergic receptor class. Now, glucagon, you just don't use the word adrenergic, but it's the same principle. Now, so the idea is these are 7 transmembrane domain proteins. They're sometimes called the serpentine receptors because they span the membranes multiple times. But the idea is these are the adrenergic class of receptors here. And there are a variety of these important receptors. But let me stress as much as I can. So many medical students, when I ask, what is the class of receptor? So many students will say it's a G protein class of receptor. Never say that. The G protein is never ever a receptor. The G protein is never a receptor. Never. They're on the wrong side of the membranes. The G protein's on the inside of the cell and the hormones on the outside of the cell. They never see each other. So the class of receptor is never a G protein. The G protein may be coupled to the receptor, but it's not the receptor. So again, it's the receptor class is not a G protein. Never ever say G protein for the receptor class. It's the adrenergic class of receptor. Okay, here it comes. Now, when glucagon or epinephrine binds to the beta adrenergic receptor class, again, there's different kinds of beta receptors, beta 1, beta 2, beta 3. But when epinephrine binds and glucagon binds to the beta receptor, the receptor undergoes a conformational change. And then on the cytoplasmic side of the receptor, it then activates a trimeric G protein called GS. And trimeric meaning 3 subunits, alpha, beta, and gamma. And because it's a G protein, it means it binds GTP in order to work. By definition, any G protein means it binds GTP in order to work. All right, here comes. Which of the 3 subunits, you must know this, which of the 3 subunits, alpha, beta, or gamma, which is the one that binds the GTP after activation? That's right, it is the alpha subunit. So the alpha subunit binds GTP, and then it undergoes a conformational change, and then beta and gamma go away. And then alpha S of GS, for the stimulatory G protein, alpha S of GS is now active because beta and gamma go away and it's bound to GTP. And then in turn, it activates another enzyme in the inner leaflet of the plasma membrane, and this one's called adenyl cyclase. Now this enzyme, adenyl cyclase, on board exams, you might see it called adenyl cyclase, or you might see it called adenylate cyclase with an ATE ending, or you might see it called adenyl cyclase with 2 YLs. It's all the same enzyme, different names for the same thing. Now, what does adenyl cyclase do once it's activated by alpha S? What it does is it takes a little bit of ATP in the cytoplasm, takes off the 2 terminal phosphates, and then it makes as a product cyclic AMP as the product. Now I'm going to show you, it's not in your book, so don't go looking for it. I'm going to show you the reaction catalyzed by, my wife will get it, sorry about that. I'm going to show you the reaction catalyzed by adenyl cyclase. Now here's the reaction catalyzed by adenyl, because sometimes if you see a structure, you remember it you remember it better. If you see a structure, you think, oh yeah, now I see what's going on here. Here is the reaction catalyzed by adenyl cyclase. Here it is. So what happens is, so again, alpha subunit of GS will activate adenyl cyclase, and what it's going to do, it's going to take this molecule at the top, and then now if you were asked, what is this molecule at the top, or what is this molecule? Well, when you look at it and say, well, it's not cholesterol, I wonder what it is. So you look at it very carefully, and you notice it has a base, a sugar, and carbon 5' 3 phosphates. Base, sugar, phosphate. Oh, it's got to be a nucleotide triphosphate. So then which nucleotide triphosphate is it? You look at the top of the molecule and you see 2 rings. You say, well, it's 2 rings, it's got to be a purine. So if it's either adenine or is it guanine? So then you look at it carefully and you say to yourself, well, at the 12 o'clock position, is there an amino group there? And the answer is yes, there's an amino group there. And if there's an amino group on a purine at the 12 o'clock position, therefore it's got to be adenine. Adenine linked to ribose and 3 phosphates, that's ATP. Look how simple that was to figure that out. It wasn't hard, it wasn't anything magical. It was a matter of thinking about it. That's ATP at the top. It was a piece of cake figuring that out. See, that wasn't hard at all. Now, what does the enzyme do? Adenyl cyclase. What it does, it cleaves this bond right here. It cleaves this bond right here, right there, cleaves that bond right there, and splits off inorganic pyrophosphate. How often have we talked about that? inorganic pyrophosphate being generated, so which is rapidly destroyed because it's toxic and it's high energy. But the idea is the remaining phosphate is then cyclized between carbon 5 prime, which it's already on, and then cyclizes it with carbon 3 prime. So adenocyclase now puts the phosphate also on carbon 3 prime, also on carbon 5 prime. Now, because the phosphate is linked to carbon 5 prime, it's a monoester linkage there. Because the phosphate's on carbon 3 prime of the ribose as well, it's a monoester linkage there. Mono here, mono there, together. It's a phosphodiester is the product. And the product is called 3 prime 5 prime cyclic, because the phosphate's in the cyclic nature. 3 prime 5 prime cyclic AMP as the product. Abbreviated cyclic AMP, but it's more appropriately called 3 prime 5 prime cyclic AMP. But remember, the phosphate's linked to 2 different carbons. It's in a cyclic nature. That's why it's called a phosphodiester linkage. The phosphate's linked to 2 di. It's linked to 2 different carbons of the ribose. Please keep that in mind in just a few minutes. So again, there's reasons why I'm showing the structure here, but the product is cyclic AMP as a product. Should you remember these details? No, nobody's going to ask you to draw the structure of cyclic AMP, but you should know what it means. Why? Because you're going to see the significance in a few minutes. You'll see why it's important to understand what's going on without the gory details. So the details, I'm not going to ask you for the structure, but you should know what 3 prime 5 prime cyclic AMP means. All right. Okay, so now once cyclic AMP is made, there it is. It can then, before we talk more about it here, let's go back to the other situation, is that when epinephrine in physiology, when epinephrine binds to the alpha-2 adrenergic receptor, again located on other tissues, but when epinephrine binds to the alpha-2 adrenergic receptor, that receptor becomes activated. It then stimulates on the inner leaflet another G protein called GI, inhibitory G protein. And then alpha I of GI binds GTP, beta and gamma go away. And then alpha I of GI then inhibits adenocyclase, because that's what the I stands for, inhibitory G protein. And then alpha I of GI inhibits adenocyclase from making cyclic AMP. Please keep that in mind, because at the end of the chapter, we're going to be talking about how does pertussis toxin work. And you must know the issue that alpha I inhibits adenocyclase from making cyclic AMP. Now coming back to the beta receptor. When glucagon or epinephrine binds to the beta receptor, it then triggers a GS to bind GTP. Alpha S then is now activated. It activates adenocyclase to synthesize cyclic AMP. Now, once cyclic AMP is made, it turns on so again, the alpha S stimulates adenocyclase, alpha I inhibits adenocyclase. But again, please remember the physiology in where these receptors are located, as in terms of which G protein is being stimulated, whether it's GS or GI here. Now, when beta receptor is triggered and GS is stimulated, and then cyclic AMP is made, then the cyclic AMP then turns on a major enzyme you got to know, and this enzyme is called protein kinase A. Now, let me stress here, from now on, now until Friday when I'm done, anytime I highlight an enzyme in the red box, anytime I highlight an enzyme in the red box, especially once we get to metabolism, automatic, high yield. Sometimes I forget to say it, But if I forget to tell you that, if it's highlighted in the red box, you got to know that enzyme. Now, what does protein kinase A do when it's stimulated by cyclic AMP? Because it's a kinase, it means it's going to use the terminal phosphate of ATP in the cytoplasm and use that phosphate to phosphorylate what? The first word says proteins. So what's going to happen is some enzymes are then phosphorylated, and by phosphorylation of certain enzymes, some enzymes are turned on and some enzymes are turned off by phosphorylation. It depends what enzyme we're talking about, whether the enzyme is on or off by phosphorylation. One more time. So again, protein kinase A will phosphorylate certain enzymes, and by doing so, some enzymes are turned on in certain pathways, other pathways by phosphorylation of enzymes. Some enzymes are turned off by phosphorylation by protein kinase A. All right? Okay, now what's coming up here is a question for you. The cyclic AMP, now once it's made by the adenylocyclase, has two fates. Two fates. One fate is to activate protein kinase A. Let me see. Now, what's the other fate not shown on this figure? What's the other fate of cyclic AMP? Let me see your answer here. Not shown on this figure. What's the other fate of cyclic AMP? What else can happen to cyclic AMP in a cell besides activation of protein kinase A? What other fate is it? Anybody? Inhibits GI? No. Good try. Good try. No. Good try. DNA? No. Yeah, there we go. See, yeah, there we go. There we go. And that is to be degraded, to be turned over. And the name of the enzyme that turns over, that degrades the cyclic AMP is a major enzyme. You talked about it in physiology, you're talking about it now, and you're going to talk about it in pharmacology as well. And that's called phosphodiesterase. Phosphodiesterase. Remember that I showed you the structure on purpose of cyclic AMP. Remember that 3' 5' cyclic AMP? I showed you because I knew I was going to talk about this. Because what the phosphodiesterase does, it cleaves the phosphate from carbon 3'. And the phosphate, it remains on carbon 5'. That's why the product is called 5' AMP as the product, because the phosphodiesterase cleaves the phosphate from carbon 3'. The phosphate remains on carbon 5', but it's no longer a cyclic molecule, and it's destroyed. In other words, it cannot activate protein kinase A. It can't activate protein kinase A. All right, now another item here, and that is you always again, cyclic AMP has two fates. One fate is to activate protein kinase A to cause certain enzymes to be phosphorylated, turning some enzymes on or off, and the other fate of cyclic AMP is to be destroyed. Now another question for you here is that you always hear about cyclic AMP as the second messenger, right? You always hear about it's the second messenger. And it is the second messenger. All right, now here comes a question for you. What is the, if that's the second messenger, what's the first messenger? Did you ever ask that question? Did you ever ask, what's the first messenger if that's the second messenger? What's the first messenger? Not the G protein, not the G protein, not the G protein. Not the receptor either. A lot of you are getting the right answer. Not the receptor complex. No, the primary message. It's the hormone. It's the hormone is the primary message being sent by the body. After all, what is a hormone in the big scheme of life? Whether it's a water-soluble or a fat-soluble hormone. What is a hormone in the big picture of life? It is a message. It's a message being sent by the body. Go tell some organ or tissue in the organ to go turn on or turn off a particular pathway. Now the lipid-soluble hormones, They're they are a primary message, but the lipid soluble hormones can get into the cell. They can go through the plasma membrane. They can come into the cell, and then once they come into the cell, they can bind to the receptor and then bind to the DNA, turn on gene expression. They don't need a second messenger. Lipid soluble hormones do not need a second messenger because they can go right through the membrane and turn on, turn off pathways directly. Whereas water soluble hormones, they are the primary message as well. But because they cannot enter the cell to turn on pathways directly, they must transduce, signal transduction, the words. They must transduce the signal that the body wants accomplished of turning on and turning off certain pathways here. So again, the hormone is the primary message, and then transduce the message to a second messenger inside the cell to go on and turn off particular pathways. Always try to understand the bigger picture. What's going on here? All right, now another item here is this enzyme, the phosphodiesterase, is always working, and it's not regulated. It's always working. It's sometimes called constitutive. In other words, it's always destroying cyclic AMP. Always destroying cyclic AMP. Well, if it's always destroying cyclic AMP, you got to ask, well, then how do you ever turn on this protein kinase A? And the answer is, it comes up in biochemistry underpinning of the physiology. How do you activate the protein kinase A if cyclic AMP is being destroyed all the time? You got to secrete the hormone. When you secrete the hormone, it binds to the receptor. It activates the GS, which in turn activates adenocyclase to make so much cyclic AMP. You make so much cyclic AMP, you overwhelm the ability of the phosphodiesterase to remove it. By overwhelming it, you then activate protein kinase A to phosphorylate certain enzymes, turning some enzymes on and off. You simply make so much more cyclic AMP, you overwhelm the ability of the phosphodiesterase to remove it. So again, it's a competition. So the idea is you're secreting the hormone, you're making so much cyclic AMP, and cyclic AMP has two fates. One is to be destroyed, which is always happening, and the other is to activate the protein kinase A. And because in the presence of the hormone, you make so much cyclic AMP, you overwhelm the phosphodiesterase's ability to destroy it. That's how you turn on this part of the pathway down at the bottom. How do you ever turn this off? You stop secreting the hormone. When you stop secreting the hormone, you stop activating the receptor, you stop activating the GS, you stop activating adenocyclase, and it's because you're not activating adenocyclase anymore, any cyclic AMP that happens to be around continues to be destroyed, so the levels drop and drop and drop and drop because it's no longer being made anymore. Eventually there'll be no more cyclic AMP, and you stop activating protein kinase A. It's a competition. So again, one more time. How do you ever turn it on? You secrete the hormone, you activate cyclic AMP, overwhelm the ability of the phosphodiesterase to remove it. And therefore you activate protein kinase A. How do you ever turn this off? You stop secreting the hormone. And by stop secreting the hormone, you're no more activating adenocyclase, no more cyclic AMP, and what cyclic AMP is around is no longer being remade, and its levels drop to nothing. There it is. There it is. That's the biochemistry underpinning of the physiology. It's a competition that goes on there. Amazing how this happens. Amazing how this happens here. All right, now on the right side of the figure, more endocrinology. Under conditions in which vasopressin is secreted or epinephrine binds to the alpha-1 adrenergic receptor. Let me ask you this. In physiology, what is the downstream purpose of triggering alpha-1 adrenergic receptor. What is the downstream purpose of triggering alpha-1? Let me see your answer here. That's right. That's right. To increase smooth muscle contraction, right? See, that's what they tell you in physiology. See, in physiology, they talk about all these hormones, and they talk about what pathways are being turned on, off, what's happening in terms of organ, tissue, increased smooth muscle contraction, blah blah blah. But that's all the end product, the final product, the final situation that occurred by that hormone. But in between is the biochemistry. What caused the vasoconstriction that went on, and by secreting epinephrine binding to the alpha-1. So the idea is if you think about this a minute, is that vasoconstriction, because you're doing smooth muscle contraction, that means ATP is being consumed in a muscle cell. And because ATP is being consumed, the muscle cell needs glucose to burn, to make ATP, to do muscle contraction. So that's what's going on here by the alpha-1 adrenergic receptor, is the generation of fuel to burn to make ATP to do muscle contraction. So how was this accomplished? So when the alpha-1 adrenergic receptor is triggered, yet another trimeric G protein is triggered in turn. This one's called GQ. Alpha Q of GQ binds GTP. Alpha Q in turn then activates yet another enzyme in the inner leaflet of the plasma membrane called phospholipase C. And what does this enzyme do? It looks for a small molecule in the membrane called PIP2. It's a small lipid in the membrane, and what it does, it does a hatchet job. It cuts it right down the middle. PIP2 is cut right down the middle, generating two fragments. One fragment is called PIP2, other fragment is called IP3. One of these fragments, IP3, will then go to the ER and then open up a channel for in the ER to allow calcium to come out of the ER, and in the muscle that's called the SCR, it's called the smooth endoplasmic reticulum. But the idea is the calcium then comes out, and then along with diacylglycerol together then stimulate yet another kinase called protein kinase C. And then protein kinase C then results in the phosphorylation of certain enzymes in the cell, in the smooth muscle cell here. So the concept here is that these three, diacylglycerol and IP3 and calcium, all three of them together, diacylglycerol, IP3, and calcium, all three together, are referred to as second messengers. Second messengers. And because the hormone is the primary message, the first messenger, and these are the second messengers because they're being stimulated by signal transduction. And therefore, these are the second messengers. Now, another item here, and that is, if they were to ask you on board exams, what are the direct activators of protein kinase C? You answer by saying diacylglycerol and calcium, not IP3. IP3 is not a direct activator of protein kinase C. It's diacylglycerol and calcium together stimulate protein kinase C. IP3 is like indirect, but not a direct activator of protein kinase C. Okay, now the idea here is now on the bottom right hand part of the figure is we're going to try to figure out what pathway is being turned on here. Again, you know, you're sending out these hormones, and a pathway is going to be turned on. But the idea here is, let me ask you this: in physiology, is you talk about when do you secrete these hormones. You say, well, because of increase in smooth muscle contraction, or you say here on the left side you secrete epinephrine because you're exercising, things like that. But as simple as I can state, what is the main reason why you secrete these hormones? And here comes the reason. Why are you secreting these hormones? It's because the blood glucose is dropping. Whether if you're exercising, as soon as you do skeletal muscle contraction, the ATP is being consumed. The muscle cell removes glucose out of the blood to burn that glucose to make ATP. And because blood glucose drops, you send out hormones called epinephrine. Or if you're sleeping in bed, you start secreting glucagon. Same thing with smooth muscle contraction. So the idea is that you're sending out these hormones because the blood glucose is dropping. So now here comes the question. On the right side over here, what pathway makes sense to you is being turned on? Not gene expression. No, good try. What pathway is being turned on? Not the Krebs cycle. Good try. And the answer is glycogen breakdown. The idea is as soon as the blood glucose is dropping out of the blood, the body responds by secreting hormones, and it's going to tell the liver, for example, go turn on glycogen breakdown to maintain blood glucose. Because we don't want to die. You know, the blood glucose is going to remember this. We talked about this on Saturday. That if the blood glucose dropped below 3.5 millimolar, because of that zero order transporter we talked about, GLUT1 and 3. If it dropped below 3.5 millimolar, that means less glucose in the blood, and therefore less glucose entering the brain. The brain's going to suffer. Coma time, and that's bad news. So the idea is the body's going to say, hey, start breaking down glycogen in not only in the liver, but also in the muscle and so on. So you start breaking down glycogen, which begins in seconds or minutes here. And again, it just makes sense. You can start rapidly breaking down glycogen within seconds or minutes. But it turns out that the amount of glycogen in the cell, in your body, the amount of glycogen in the human body is limited. Not now, but later on, I'm going to ask you, how long does your glycogen last? Don't answer right now, don't answer. But somewhere along the line, I'm going to ask you, how long does your glycogen last? You do not have an infinite supply of glycogen. You do not have an infinite supply of glycogen, but it will run out sometime. And so the body says, okay, well, we know it's going to run out. So therefore, we got to start kicking in another pathway because the blood glucose will drop and will die. So the body says we got to kick in another pathway while the glycogen is running out. And sure enough, what happens is this same enzyme in the liver, protein kinase A, what it's going to do is going to look for a specific transcription factor called Krebs, which is normally inactive. And then by phosphorylating it, it's now active. And again, Krebs stands for cyclic AMP. Notice it's responding to the cyclic AMP. Cyclic AMP, response element binding protein. And when it's phosphorylated, it's active. It then goes into the nucleus of the hepatocytes, looks for a piece of DNA called the CRE, which is an enhancer region, which is it's an enhancer for a major gene in the pathway called gluconeogenesis. And sure enough, that gene will be turned on by RNA polymerase, and then as a result, the pathway of gluconeogenesis is turned on, and you start synthesizing glucose. So the concept here is that this Krebs protein is, we already talked about this. We have already reviewed this earlier. Where did we first talk about this? Where did we first talk about turning on gene expression for gluconeogenesis? And you're all going to say right here, page 78. Page 78. This is where, just look at that page. You'll see this part of the figure right here has already been reviewed. How you're turning on gluconeogenesis at the gene level here. All right, now the concept here is, now what happens, so you start turning on gluconeogenesis, and because it's taking hours, because it's being done at the gene level here. All right, and I got a question for you here. Is not a question per se, but the idea is, what happens is a lot of students think what the body is doing is that when the blood glucose drops, the body first breaks down glycogen and then kicks in gluconeogenesis. In other words, sequentially. You first break down glycogen and then you kick in gluconeogenesis. There's no way that can happen sequentially. You'd be dead if it happened sequentially, where you first did glycogen breakdown and then you did gluconeogenesis. You'd be dead. Why would you be dead? Look at it. Look at it. Because glycogen, when it runs out, if the body says, well, okay, now I got to kick in gluconeogenesis, because it takes hours to kick in. When glycogen is gone, the blood glucose continues to drop and drop. And then you say, well, okay, I got to kick in gluconeogenesis because it takes hours. The blood glucose will drop to more and more and more. You'd be in a coma and you'd be dead. It cannot happen sequentially. You'd be dead. What I'm driving at is that this is happening simultaneously, concurrently. While you are breaking down glycogen, simultaneously you're kicking in gluconeogenesis, such that when glycogen is gone, and it will be gone, gluconeogenesis better be at its maximum, keeping me and you alive. And it will be at its maximum when glycogen is gone, keeping me and you alive. And how can this occur simultaneously? Notice the same enzyme, protein kinase A, is kicking in glycogen breakdown and simultaneously kicking in gluconeogenesis. Beautiful, beautiful how this works. While you are breaking down glycogen, simultaneously you're kicking in gluconeogenesis. You must understand they are occurring at the same time. And you must understand you'd be dead if it occurred sequentially, not simultaneously. You can't just memorize that. You have to understand why it has to occur simultaneously here. They go on together. Now, one more item here. One more item. And that is okay, everybody with me here. You must understand this. This is part of endocrinology and biochemistry here. Now I got a little oral vignette here, oral vignette. Suppose they say to you that tomorrow morning you wake up, and when you wake up, you measure your blood glucose and you find out it's a certain value. And then the day after that, when you wake up, before you measure your blood glucose, you go to your kitchen and you make and drink a cup of black coffee. No sugar, no cream in that coffee. And then after you drink that coffee, then you measure your blood glucose and you find out, wow, look at this. My blood glucose is higher than it was the day before when I didn't drink that coffee. How do you explain that? Obviously has something to do with this page, right? It has something to do with this page. Why did your blood, so here comes the question. Why did your blood glucose rise when you drank that coffee? Or I can change one word instead of saying coffee. Why did your blood glucose rise when you drank that tea? That black tea, no sugar, no cream in that tea, yet your blood glucose would rise. What's it got to do with this figure? Okay, that's right, there's something in that coffee. There's something in that coffee or tea. And what is it in pharmacology? What's that class of molecules called? The class of molecules I know it's caffeine, but what's the class of molecule called? What's the class of molecule called? Stimulants, agonists. That's too. There we go. Now we're getting close. Semanthe. That's right. They're called the methylxanthines. The methylxanthines in pharmacology. And what do the methylxanthines do? Why did your blood glucose rise? And the answer is right here. Because the methylxanthines, what they do is they inhibit the phosphodiesterase. That's what they do. And so now, why did your blood glucose rise? Because here it is. Because when you drink that coffee, when you wake up in the morning after sleeping, you're secreting glucagon. Glucagon is activating its receptor, activating GS, activating amino cyclase to make cyclic AMP. And a portion of the cyclic AMP, when you wake up in the morning, is supposed to be inhibited by the phosphodiesterase. But now if you inhibit its destruction, what happens to the cyclic AMP? It starts to rise. One more time. And so if it starts to rise, more protein kinase A, more glycogen breakdown, more gluconeogenesis, and your blood glucose would rise. You'd be hyperglycemic. Okay. One more time. By drinking coffee or tea, you're inhibiting the phosphodiesterase when you wake up in the morning. And therefore, some of that cyclic AMP is supposed to be destroyed, but now it's not being destroyed because the PDE is being inhibited. Therefore, cyclic AMP levels rise higher than normal. If it rises higher than normal, more protein kinase A, therefore more glycogen breakdown, and therefore more gluconeogenesis. That's why you become hyperglycemic. Here, it makes sense. Now, if it doesn't make sense, find out why. It must make sense to you. If it doesn't make sense, don't be stubborn. Find out why. Use the Q&A pod. Dr. Prashad and Dr. Mahad, they'd be happy to answer the question, but it must make sense. You should be able to verbalize. Why is it when I drink a cup of coffee with no sugar in it, yet my blood glucose would rise? You should be able to verbalize the answer. Major integration of biochemistry with physiology and pharmacology here. All right. Okay, now what's coming up next? That's right. Kitsia. That's right. It makes sense. Eri. That's right. There we go. It makes sense. No excuses. If it doesn't make sense, find out why. I want you to know why. Here. Must know why. Now what's coming up next is the second of the three major signal transduction pathways, also discussed in physio and certainly going to be discussed in pharmacology. Now this is the issue of signal transduction to cause vasodilation. Now on the left, now suppose this represents a smooth muscle cell. Under conditions of distension, when the person secretes the hormone ANF. Now, by the way, you might hear it called ANF or you might hear it called ANP, but it's one and the same. This is the atrial natriuretic factor or peptide, same thing. So ANF or ANP is one and the same. It's a hormone that's being secreted under conditions of distension. Now, when the hormone is secreted, it's going to bind to its receptor. Now notice here the receptor is not a serpentine receptor, not a 7 transmembrane domain. It spans the membrane one time. But when the hormone binds to its receptor, then the receptor in the smooth muscle cell becomes activated, and then on the cytoplasmic side, it'll take a little bit of GTP and then convert it by taking off 2 phosphates as 3', 5' cyclic GMP as the product. So again, in the smooth muscle cells, you're generating cyclic GMP inside the cell by the action of the hormone via its receptor. Okay, now on the right side of the figure, major integration with physiology and pharm. Under conditions in which you secrete vasodilators, what they're going to do is they're going to turn on a major enzyme in the vascular endothelial cells called nitric oxide synthase. Now, what does this enzyme do? It tells you what it does. It synthesizes nitric oxide using the substrate arginine. We talked about this on Saturday. One of the nitrogens of arginine is cleaved off. It's then made in nitric oxide as the product here. All right, now before we move on the upper right-hand side of the page, here comes the pharmacology. Now get ready with your responses. Under what conditions do you secrete nitroprusside? Under what conditions do you administer? What pathological or clinical problem do you administer nitroprusside to treat? Well, it's mostly used for hypertension purposes. Hypertension. Now, nitroglycerin would be used for angina. As when people said angina, so most often nitroglycerin would be used for the angina situation. What about isosorbide dinitrate? Dinitrate also would be used for angina. That's right, Marcelo. That's right, for angina purposes. Now notice here that these three pharmacological drugs are all nitrogen-containing drugs. So when they are administered pharmacologically, biochemically, they are then converted to nitric oxide here. So again, at the upper right-hand part of the page, look at the major integration of physiology on the left that Dr. Wilson talked about. On the right side, pharmacology that Dr. Harris will talk about. And I'm talking about regarding nitric oxide here. Major integration of three disciplines. Now, once nitric oxide is generated, fortunately, it's permeable to the membrane. There's no receptor for it. Fortunately, because its half-life is seconds. It sticks around only for a few seconds. It goes right through the membrane and looks for an enzyme called guanyl cyclase. And what does guanyl cyclase do? What does it do? Just like adenyl cyclase on the previous page, guanyl cyclase in the smooth muscle cell takes a little bit of GTP, takes off the two terminal phosphates, and makes 3' 5' cyclic GMP as the product. Notice here on the left side how ANF administration generates cyclic AMP, and so does nitric oxide generation also generates cyclic GMP. Now, once cyclic GMP, let me finish off this figure, then we'll take the break. Once cyclic GMP is formed, it turns on a major kinase called protein kinase G. Here, now before we move on, now don't get these second messengers because cyclic GMP is yet another second messenger. Don't get these second messengers and what kinase is being turned on confused. It's not that hard. Here it is. Here it is. This is what you must know. All you need to know is cyclic AMP turns on protein kinase A. Cyclic GMP turns on protein kinase G. And calcium turns on protein kinase C. Which second messenger is turning on which kinase? Okay, not hard. Not hard. All right. Okay, so again, you understand the concept here. You must understand the concept. Which second messenger is turning on which kinase? Who's zooming who? All right, now to finish this figure off. Now there's a little dilemma which I would rather have you figure out first. Because notice here, a kinase is being turned on. Now a lot of students won't get this right, so I want you to try to think about this. A kinase is being turned on, yet vasodilation is the outcome. So now we got to try to figure out, well, if a kinase is being turned on, that must mean, therefore, by smooth muscle that causes contraction. Let me see the answer. What's the name of the protein that causes contraction? What's the name of the protein that causes contraction in vascular smooth muscle? Shagrin, a little bit more detail. Name of the protein that causes contraction. Not myosin, which part of myosin? And it's called, here comes the answer, and it's called myosin light chain. Myosin light chain causes contraction. Okay, but now let me ask you this. Does myosin light chain cause contraction in the phosphorylated state or in the dephosphorylated state? Let me see your answer. Does myosin light chain cause contraction in the phosphorylated state or in the dephosphorylated state? Protein kinase G to cause the vasodilation. There we go. Ahmad, anybody else out there? That's right. Without looking at Ahmad's answer, Samantha, that's right. Everybody, that's right. That's right. That's right. And the answer is, it's got to be a phosphatase, right? It just makes sense here. So now here it is. I'm going to say a sentence to you right now that sounds very convoluted. Very convoluted. But listen carefully, because this is exactly what happens. The straightforward biochemistry events. Here it comes. Here it comes. Ready? Everybody ready? I'm going to tell you the biochemistry of what's happening. Here it comes. The activated protein kinase G causes phosphorylation of a phosphatase. The phosphorylated phosphatase is now active. The phosphorylated phosphatase then causes dephosphorylation of the phosphorylated myosin light chain. The dephosphorulated myosin light chain cannot cause contraction, therefore relaxation occurs. Now I know I said that very rapidly on purpose. I didn't stay up last night practicing that words. You can say the same thing as me. I'm going to say it one more time, a lot slower. But you can say the same thing as me by just verbalizing the answer several times. Now here it is. This is the sequence of events, much slower the second time. Here it is. The activated, now just look at the bottom of the figure and watch what happens. The activated protein kinase G causes phosphorylation of a phosphatase. The phosphorylated phosphatase is now active. The phosphorylated phosphatase then causes dephosphorylation of phosphorylated myosin light chain. The dephosphorylated myosin light chain cannot cause contraction, therefore relaxation occurs. Just like that. Just like that. Now again, while it's fresh in your mind, later on tonight or whatever, just practice the same verbiage. Look at this, then if you think you mastered it, look away and verbalize the same answer. Just verbalize the answer. Just try it on your own and you'll get to the same answer. You can't stare at this and expect to be able to spit it back later on a board exams. You have to practice this several times, and then you'll be just like me, just like that. Okay, just try it out. Just try it out. Okay, now one more item here. Then we'll take the break, and that is the major integration with pharmacology. Now, in pharmacology, at the break or tonight on TV. If I go watch a basketball game, what happens is I go watch a basketball game on TV, the NBA, after we're done tonight, and then there'll be some commercials on TV, and in these commercials on TV, there might be a man and a woman sitting in the bathtub, and they're smiling at each other, and then a commercial will say, when the time is right. Okay, and now what's the name of that drug that's has something to do with this page? That's right. That's right. It is the sildenafil, Viagra, and so on. So now, what do they got to do with this page? And it has to do with the idea of the cyclic AMP. Now, remember on the previous page, cyclic AMP is always destroyed by a phosphodiesterase. Same thing here. The cyclic GMP is always being destroyed by yet another phosphodiesterase in the smooth muscle cells. And again, this phosphodiesterase is always working, always destroying cyclic GMP. And how do you ever turn this on? You secrete nitric oxide or you secrete ANF. You overwhelm the ability of the phosphodiesterase to remove it. And how do you ever turn it off? You stop secreting the hormones. And once you stop secreting the hormones, then cyclic GMP is no longer being made, and the phosphodiesterase will destroy it. But now the idea is, If somebody takes Viagra, then what the drug does, it inhibits the phosphodiesterase, so the cyclic GMP is not being destroyed. And as you know, the cyclic GMP, if it's not being destroyed, the levels start to rise. And when it starts to rise, you know the big picture in more ways than one. But you know where I'm driving at here. You know the rest of the story with that stuff there. But there it is. That is the idea. That's the target of the phosphodiesterase. All right, now let me ask you this again. So the idea is by inhibiting the phosphodiesterase, the cyclic GMP is no longer being destroyed. And if it's no longer being destroyed, look at all that relaxation because it activates protein kinase G, activates the phosphatase. The myosin light chain is always dephosphorylated, causing all that relaxation. And you know where that's going on, all that relaxation that goes on. All right, one more item about this. On those commercials on board exam, on those commercials on TV, if you listen very carefully, the announcer will say that anybody taking a drug like sildenafil should not, you might say, anybody taking an antihypertensive drug should not be taking sildenafil. No, because you could die. Now, why is that? Why is it bad or could be lethal by taking a hypertensive drug at the same time taking Viagra? Why could that kill you? That's right. Just look at the biochemistry explains it. There it is in front of you. Because if you take a hypertensive drug like nitroprusside and you generate so much nitric oxide, generating so much cyclic GMP, and a portion of it is supposed to be destroyed by the phosphodiesterase. But if not, if you're inhibiting the phosphodiesterase with sildenafil, then the cyclic GMP is so high and therefore so much relaxation, the blood pressure, the smooth muscle blood pressure then would drop too severely and you could die under those circumstances here. There it is, how the biochemistry explains the pharmacology. Everybody with me? Everybody with me? It makes sense. And if it doesn't make sense, find out why. All right, so ask. Okay, so right now it's 4 minutes after the hour. I'm sorry I went a little bit, but I wanted to finish off this figure. So let's take a 10-minute break, and then I will be back. See you shortly. Okay, welcome back to hour two. Now, before we leave this particular figure in front of us here on relaxation, now this is where I want you to. And by the way, Samantha did terrific. What you typed out there in the public chat pod is exactly the description, the wording that would be used. She did, that was perfect. See, but notice she knows the material and she can spell it out. She can either verbalize or write it out without looking. Okay, now it's the same thing here on this diagram on page 138. Just look at this and just stare. Just look at this and try to verbalize what's happening. Now, without looking at what Seaman wrote, which is perfect answer. Look, you could say the same thing. Just look at this for one more time. Protein kinase G causes phosphorylation of a phosphatase. It's now active. The arrow is up, which then in turn dephosphorylates the phosphorylated myosin light chain, and by dephosphorylating phosphorylated myosin light chain without the phosphate group on, it cannot cause contraction. Therefore, relaxation occurs. That's exactly what Samantha wrote, and you could say the same thing by looking at this particular diagram. Okay, it's nothing hard. It's just a matter of going through this, and then once you think you know this, then put it aside, look away, and try to verbalize the same thing. Try it out. Just try to verbalize the answer, and if you make a mistake, go back and do it again until you mastered it. Picture what's happening. It's part of the learning process. You can do it. You can do it. Give yourself credit, but there's no substitute with active learning. You got to want to know this. As long as you want to know this material, just like Samantha did, you're going to do the same thing. So just do that. You'll learn it a lot better here. Okay, just try it out. I mean, there's nothing magical. There's nothing magical here. You're either medical students or doctors already, and you should want to know this information. Okay, now what's coming up next is another third and last of the signal transduction pathways, extremely high yield, and that is how insulin works and how growth factors work. Now, you should want to know this, right? You should be eagerly saying, I can't wait to hear this because I want to know how insulin works. I want to know how growth factors do their thing, right? You should want to know this because there's a major integration with pathology coming up here, so as well as endocrinology. So you should want to know this information, and it's extremely high yield. Okay, so here it is. This is now the how growth factors and insulin mediate signal transduction. So everything I'm going to talk about is in reference to insulin, but growth factors work basically the same way. The target organ is different, growth factors versus insulin, but the intermediate biochemical steps are basically the same. Okay, so when you secrete insulin under conditions of hyperglycemia, insulin will go to certain tissues. Now, not now, but later on, I'm going to ask you, what are the insulin-dependent tissues? There aren't that many. But later, I'm going to ask you, what are the insulin-dependent tissues in the human body? There aren't that many. But once insulin finds its receptor, insulin will dock with its receptor, and the receptor dimerizes. That's the first thing that happens. Two monomers of the receptor come together in the presence of insulin. And then once the receptor dimerizes, the receptor now becomes activated. And it turns out the receptor also is an enzyme. And the enzyme activity it possesses is called tyrosine kinase activity. It has tyrosine kinase activity. So sometimes you hear it as a tyrosine kinase receptor for insulin. So on the cytoplasmic side of the receptor, after the receptor dimerizes, because it has tyrosine kinase activity, what does it do? Because it's a kinase, it's going to take the terminal phosphate of ATPs from several ATPs in the cytoplasm and puts those phosphate, now the first word says tyrosines. And so now the question is, so it's putting phosphates onto target tyrosines, because that's what a tyrosine kinase does. It puts phosphate from ATP on the target tyrosines. And now the issue is, where are the target tyrosines? And it turns out its own tyrosines. It puts on phosphates onto itself, onto its own tyrosines. That's why it's called autophosphorylation of the receptor. One more time. In the presence of insulin, or if this is a growth factor, like epidermal growth factor, what happens is when the hormone binds to the receptor dimerizes, it becomes activated. On the cytoplasmic side of the receptor, it puts phosphates onto itself. It autophosphorylates itself. But now the question is, why did it autophosphorylate itself? Do you think it autophosphorylated itself so that the receptor can say to all the proteins in the cytoplasm, hey, proteins in the cytoplasm, look at me over there, look at me, I have all these phosphate groups on me, I am nice and pretty, and you're not? Or do you think it's doing it for a purpose? That's right, it's doing it for a purpose. Why do you think it puts phosphates onto itself? For what purpose is that? And the purpose is to attract a protein in the cytoplasm called IRS1, which stands for insulin receptor substrate. So this is another protein in the cytoplasm that now will dock with the autophosphorylated receptor. It will never dock unless the receptor has phosphates on it. So IRS1 then docks. So I'm taking this part of the figure right here and moving it on the right side with IRS1 now docked. And then what happens? A number of other proteins then dock in turn. It's like a cascade, signal transduction. And this class of other proteins that dock are all called SH2 domain proteins. Don't worry about what that means, low yield. But one of the SH2 domain proteins is called PI3 kinase. This is a medium yield enzyme that's located in the adipocyte and muscle, and this is the enzyme that when it's activated, it then allows GLUT4. We'll talk about it later on. GLUT4 is put onto the surface of the adipocyte and muscle here. Notice the cascade, how insulin turns on its receptor, which attracts IRS1, which then attracts PI3 kinase, which then opens up a vesicle to allow GLUT4 to be put onto the surface here. Amazing how this happens. Signal transduction. The primary message is the insulin, the hormone here. Now yet another SH2 protein can dock. Now the name of this one is not important. And after a number of steps that you don't have to know, including the activation of RAS, then downstream certain specific transcription factors are turned on. And these specific transcription factors then turn on genes involved in cell division. If this were growth factors, the genes are involved in cell division. If this was insulin, these would be genes involved in fatty acid synthesis, for example, cholesterol synthesis. It depends which hormone on the upper left-hand part of the page we're talking about in terms of what genes are being turned on. It's a signaling to turn on genes depending on which hormone we're talking about here. Okay, now question for you. What class of protein is RAS? What class of protein is it? Is it a kinase class? Is it a receptor class? Is it a phosphatase class? What class of protein is RAS? MAP kinase. What class of protein is RAS? Not kinase. No, not kinase. No. And there we go. Now we're getting it. And the answer is it's a G protein. Now, how do you know it's a G protein? Because look at the figure. Right underneath the RAS, it says G protein. RAS is a G protein, but it's a monomeric G protein, P21. It's a monomeric G protein in contrast to the trimeric G proteins that we talked about earlier, like GS and GI and GQ. This is a monomeric G protein, one subunit, and it must bind GTP in order to work. And once RAS binds GTP, it then goes on and turns on specific transcription factors, and then certain genes are then turned on. Now what I'm going to do right here on the upper right hand part of the page, what I'm going to do is I'm going to outline what I just said. Just sequence of events I just said. What I said is that when I secrete insulin, or when you secrete insulin or a growth factor, in turn, this is going to activate a tyrosine kinase receptor. And depending on which hormone we're talking about, but it's a tyrosine kinase receptor class is being turned on, which in turn attracts IRS1 and activates it, which in turn activates SH2 proteins, and then downstream activates RAS, which in turn downstream activates specific transcription factors, which in turn downstream activates and turns on certain genes here. Notice now the sequence of events. And notice here that these are all proteins. And none of these proteins should work unless the preceding proteins are activated. Now what I mean by that is, for example, RAS should never bind GTP and turn on specific transcription factors to turn on gene expression. RAS should never bind GTP unless SH2 proteins dock with the IRS receptor, and it never docks with the tyrosine kinase receptor unless the receptor autophosphorylates itself, and it never autophosphorylates itself unless the receptor dimerizes, and it never dimerizes unless you secrete the hormone. So none of these proteins work unless the preceding proteins are turned on, and they're never turned on unless you secrete the hormone. This is all normal. Now, importantly, the class of genes that encode these normal proteins, they're classified as proto-oncogenes. Proto-oncogenes are normal. They are normal genes. Proto-oncogenes are the class of genes that encode these normal proteins. Proto-oncogenes are normal genes that encode these normal proteins. Now, as we talked about earlier last week, any gene can be mutated. And if the proto-oncogene for any of these proteins gets a mutation in it, for example, if the proto-oncogene for RAS, which is normal, gets a mutation in it, such a missense mutation, something like that, such that the protein is still being made, but now because of the mutation, the RAS protein binds GTP in the absence of the preceding steps. If it binds GTP in the absence of the hormone being secreted, for example, then RAS will then activate specific transcription factors in the cells, and then causing certain genes to be turned on. If these genes are involved in cell proliferation, the cells will start to grow and grow and grow and grow. And now that proto-oncogene that got a mutation in is now classified as an oncogene. An oncogene now makes a defective protein that now works when it should not work. And that's what oncology, a lot of that of oncology is about. All those oncogenes that Dr. Barone is going to be talking about next week in oncology, the concept is an oncogene it's a proto-oncogene, which is normal, that got a mutation in it, such that the gene is now called an oncogene, and it makes a protein when it works when it should not work. Any of these proto-oncogenes can be mutated. For example, if the proto-oncogene for the tyrosine kinase receptor gets a mutation in it, such that it autophosphorylates itself in the absence of the hormone, if it autophosphorylates itself inside inside the cytoplasm, if it puts phosphates onto itself in the absence of the hormone, if it puts phosphates onto itself, IRS1 will now dock, SH2 proteins dock, RAS becomes activated, then specific transcription factors are turned on, genes are turned on. As a result, cells grow when they should not grow. And that gene, in this case here, the tyrosine kinase receptor, is now called an oncogene. So all those oncogenes that Dr. Barone is going to talk about, like Herb and Mick and June and this and that, and RAS and so on, is simply a normal proto-oncogene that gets a mutation in it, and it works when it should not work. There it is. There it is, how the biochemistry explains the oncology here. All right, now continuing on with insulin. Okay, I'm making it simple. There it is. That's what an oncogene is. Again, please understand, we talked about it last week, but please understand the difference between a proto-oncogene and a tumor suppressor gene. They're two different things. Last week we talked about tumor suppressor genes causing cancer. Remember all that? Leaf-Fermenting syndrome. Remember the cell cycle. Remember all that? You know, we talked about that last week. But tumor suppressor genes, they work differently than the proto-oncogenes that we're talking about right now. But you must understand. You got to understand. You got to learn this stuff. You can't memorize. You should want to know this stuff. You should want to understand this stuff. It's not hard. It's not hard. Just want to understand this stuff here. Okay, it's good. It makes sense perfectly. All right. Okay, good. Now the idea here is that continuing on is coming back to the normal situation with insulin. So when insulin finds its receptor, normally it dimerizes, autophosphorylates, and then it tracks IRS1, and yet another SH2 protein can dock that you don't have to know. And then after a number of steps you don't have to know, don't worry about them, insulin downstream. Remember, insulin never enters the cell, never, ever enters a cell. But inside the cell, downstream, because of the action of insulin, can activate a phosphatase. And the phosphatase, when it's turned on, will dephosphorylate certain enzymes. And by dephosphorylating certain enzymes, some enzymes are turned on and some enzymes are turned off by dephosphorylation by insulin mechanisms here. Now this part here in the lower left side of the figure, major concept here. Here comes the major concept with endocrinology integration. Remember several figures ago, we talked about the idea is that when you secrete glucagon, glucagon results in increased phosphorylation of certain enzymes. Remember all the issue of adenocyclase making cyclic AMP to activate protein kinase A. And protein kinase A phosphorylates certain enzymes, turning some enzymes on and off. Depends which enzyme we're talking about. But the phosphate doesn't remain on the enzymes forever under conditions in which you secrete insulin. Insulin results basically of this dephosphorylation of the same enzymes that glucagon resulted in the phosphorylation of here. So insulin results in dephosphorylation basically of the same enzymes. And those enzymes are going to be on or off. It depends on what enzyme we're talking about here. Now what I want to do here is illustrate how important this is. Phosphorylation, dephosphorylation. How critical this is to the human body. The mechanism of phosphorylation, dephosphorylation under the command of hormones. Now what I want to do here is illustrate this in the context of glycogen metabolism. Now we'll talk about glycogen metabolism, the details later on this week. Don't worry about the details right now. But the concept I'm trying to get across here is in terms of glycogen synthesis and breakdown. Now in the body right now, if I were to ask you where do you break down glycogen and make it in the body, you say the liver and the muscle. The liver and the muscle are the two major organs that make and break down glycogen, the muscle and the liver. All right, now in the liver and the muscle cells, in the cells. Where is the pathway of, let me see your answer here. Where is the pathway of glycogen synthesis? Where is it? Where is the pathway of glycogen synthesis? You should know by the way, let me stress here, before, you got to know where things are happening inside cells. Where is the pathway inside the liver? Where is it occurring? Is it occurring in the mitochondria? Where do we make glycogen? Is it in the mitochondria, nucleus, cytoplasm? Where are you making glycogen? That's right. It's in the cytoplasm. And the top enzyme for making glycogen, you cannot ask for a better name. It's called glycogen synthase. Synthesizes glycogen in the cytoplasm. All right, now in the liver cell. So you make glycogen in the cytoplasm because that's where glycogen synthase, the enzyme, is located. Now, where in the liver cell do you break down glycogen? We're working out. Now, again, you got to know where is glycogen broken down in the liver cells. By the way, let me stress here. Before we ever get to metabolism, coming up later on this week, before we ever get to it, there are 3 things you need to know about metabolism. 3 things. Location, location, location. You got to know those 3 things. If you want to understand metabolism, if you want a grasp of metabolism, to understand it, to appreciate metabolism, you got to know location, location, location. You got to know what organs and tissues do this or that, and inside those tissues, inside the cells, where is a particular pathway occurring, as illustrated right here. You must understand where things are happening. Location, location, location. So where in the liver cells is glycogen broken down? It's synthesized in the cytoplasm, but where is it broken down? Let me see your answer here. Where is it broken down? SES, cytoplasm. Give everybody a chance to write. Cytoplasm, mitochondria. Now here's a way you get to the answer. Here's a way I can give you the answer real quick, but I'd rather have you think about it. Now, if you're thinking mitochondria or ER or some other organelle, there's no way. Now think about it. Glycogen is a huge hydrophilic water-soluble particle with all these glucoses in it. And if it's made in the cytoplasm, and you need to break it down, there's no way you can move it into the mitochondria or some other organelle. You got membranes to move across. Remember, the mitochondria has 2 membranes. You can't move it into the peroxisomes or the mitochondria or the nucleus because it has membranes. And so what I'm getting at, and there's no way you can move a big glycogen granule across membranes to break it down. So what I'm driving at is that glycogen breakdown also occurs in the cytoplasm. And the top enzyme for breaking down glycogen in the cytoplasm is called glycogen phosphorylase. Major enzyme we'll be talking about later on this week. Major diseases called McArdle's disease, HERS disease, glycogen phosphorylase. Now here comes the concept here. Now just picture, in the cytoplasm of the liver and the muscle are these 2 major enzymes. 2 major enzymes of the respective pathways. Glycogen synthase, which synthesizes glycogen. Glycogen phosphorylase, which breaks it down. And they're sitting right there in the cytoplasm together. They're in the same compartment in the cytoplasm of your liver and the muscle. And they're sitting there. Just picture these 2 enzymes. They're dominant enzymes of the respective pathways. They're sitting there, thinking, looking at each other, thinking, who's gonna blink first? So glycogen synthase says, well, I'm gonna make glycogen. And then glycogen phosphorylase says, you make it, I'm gonna break it down. And then glycogen synthase says, well, you break it down, I'm gonna make it, you make it, I'm gonna break it down. It's like 2 kids fighting with each other. So the body says to these 2 enzymes, just like in the movie Austin Powers, behave. And so now how is the body going to make these 2 enzymes behave? And here comes the answer. By phosphorylation, dephosphorylation. Right here. Here it is. Get rid. The integration with endocrinology and biochemistry once again. Now here are these two dominant enzymes of the respective pathways, glycogen phosphorylase and glycogen synthase. It makes no sense that these two enzymes work at the same time. It would be futile. So how are these two enzymes controlled? How are they made to behave? And the answer is, here it is, the idea of hormone, how hormones direct the show. How the hormone glucagon, when you secrete glucagon, now see this little arrow right here, this black arrow right here, see that little black arrow? We had a whole figure on that arrow, a whole figure on that arrow. How glucagon results in the activation of protein kinase A. Protein kinase A, what it's going to do is take the terminal phosphate of several ATPs. One of the phosphate groups is removed from ATP and put onto glycogen phosphorylase. Another is put onto glycogen synthase, and it just so happens that phosphorylated phosphorylase is active, phosphorylated synthase is inactive. As a result, you break down glycogen and don't make it. Beautiful how that works. All because glucagon was secreted. But then, these phosphates don't sit here forever on these enzymes. Under conditions in which you secrete insulin. Now see this little black arrow here under above insulin here. See that little black arrow? We had a whole figure on that arrow. That was the previous page. A whole figure on this arrow. You know the details already. And so when you secrete insulin, how insulin downstream activates a phosphatase. And what does the phosphatase do? It pops these phosphate groups off. They come off. And when they come off, then we get dephosphorylated phosphorylase, and it turns out it's inactive. Glycogen synthase without the phosphate groups on are active. As a result, glycogen is made and not broken down. Beautiful how this works. Phosphorylation, dephosphorylation. How some enzymes are on and off by reciprocal phosphorylation. This is incredibly beautiful how the body works here. This is just glycogen, but this is going to be applicable to other things coming down the road, triglycerides, cholesterol, so many things. Phosphorylation, dephosphorylation. Beautiful, under the command of the hormones. Now what you're looking at right here, is the third and last of the ways the body chooses to regulate a particular pathway. Third and last way. Remember, the first way was at the gene level. Any pathway being turned on at the gene level is very, very slow. We talked about that way back on page 78. And then the second of the three ways we talked about on Saturday, that was allosterically. You can turn on and turn off enzymes and therefore pathways by allosteric agonists and antagonists, which takes place within seconds. You can turn on and turn off enzymes and therefore pathways. And the third way the body can choose to turn on and turn off enzymes and turn off pathways is by phosphorylation, dephosphorylation, which also within seconds. You can turn on, turn off pathways. How fast is phosphorylation, dephosphorylation? If this were epinephrine at the top of the figure here, epinephrine. If I were to walk down the street at night, and out of the bushes comes a pit bull in my direction, and this pit bull, the owner is Michael Vick, and Michael Vick didn't feed that pit bull. Pit bull is hungry and wants to take a chunk out of my leg for fresh meat. And if I have a desire to run, I got to run as fast as I can. And to run, I need ATP. And if I need ATP, I need glucose. And where is glucose? It's in glycogen. So I got to make glycogen break down rapid. How rapid is it? As soon as I see that pit bull, as soon as I turn my eyes and I see a pit bull charging at me, I secrete. I start heating up. Sometimes called the adrenaline rush. I start to heat up because I'm secreting insulin. Why? Because my muscle cells are removing glucose out of the blood. My muscle cells are breaking down glycogen within seconds, within microseconds. I'm secreting epinephrine. Epinephrine's carrying out this black arrow at the top, activating protein kinase A, and protein kinase A phosphorylase to phosphorylase. I break down glycogen within seconds and don't make glycogen because I'm phosphorylating the synthase enzyme here. You break down glycogen within seconds and don't make it. How beautiful this works. Now, if you're sleeping in bed, you're not secreting epinephrine, so you're secreting glucagon. This is not going on within seconds, but the idea is this could go on within seconds or microseconds. Phosphorylation, dephosphorylation, how rapid this is. The third and last way the body chooses here. All right, major concept. Major concept. You're going to see it again and again and again in metabolism. Phosphorylation, dephosphorylation. The sooner you master this, the better off you're going to be. All I can say, the sooner the better. All right, now the last item we're going to talk about here, the last thing we're going to talk about is of this chapter is G protein dysfunction here. Now in G protein dysfunction, what this is about is remember G proteins are involved in on that one figure we talked about regarding glucagon epinephrine. Remember all that page of the beta receptor and all that kind of stuff. So the idea here is, but now this is where something can go wrong with the G protein. Now, earlier last week, we talked about ADP ribosylation. We know what that means. We know what it means here. But the idea is, what was the substrate? Let me see your answer here. Everybody should tell me the answer. What's the substrate for these toxins using that do ADP ribosylation? What's the substrate? Everybody should be saying, yeah, that's right. Thank you, thank you, thank you, thank you, thank you. Hey, Vio, don't worry about the other stuff about that movie stuff. I didn't ask that. What was it, American Children movies? I'm talking about what was the name of the substrate. And you should, Vio, you should be saying NAD, not something about some movie show. Got to be paying attention. Okay. All right. Now the thing here is, so it's NAD. Now the concept here is that when you already talked about this with Dr. Muscatello, but the idea is that under conditions in which cholera toxin or E. coli toxin are secreted by the pathogenic organisms, what they're going to do is ADP ribosylate alpha S of GS in both of these situations here. And as a result of ADP ribosylation of alpha S, cyclic AMP levels rise with cholera, and same thing with E. coli toxins. Cyclic AMP levels rise. Okay, now why? And then as a result of cyclic AMP levels rising, then it opens up channels in the mucosa for salts. You lose a lot of salt, and that's severe diarrhea that occurs in cholera, as well as the traveler's diarrhea. All right, now we got to explain the why. Why did the cyclic AMP levels rise by ADP ribosylation of alpha S? All right, now to explain the why is you have to know the normal biochemistry first. And now earlier in the first hour, if I were to ask you, what is alpha S normally supposed to do? And you would say, because of the little s, alpha S is supposed to stimulate adenocyclase to make cyclic AMP. And it does. But now, why did the cyclic AMP levels rise higher when alpha S is ADP ribosylated by cholera toxin? What do you think happened? What do you think? Why did the cyclic AMP levels rise higher? What do you think happened? That's right. There you go. That's right. Not that you're inhibiting GI. No, it has nothing to do with GI. GI is in the wrong tissue. That's right. You're stimulating the stimulator. Alpha I is supposed to stimulate adenosylase to make cyclic AMP? Yes. But by putting ADP ribose on it, it works even better. It works even better. And so it stabilizes the GTP, and it works even better, and then adenosylase makes more cyclic AMP. There it is. Not hard, just to understand what goes on. Now, pertussis toxin, it's a little bit more difficult to figure out. But pertussis toxin works by ADP ribosylation of alpha I, inhibitory, alpha I of GI. And as a result, cyclic AMP levels are affected. All right, now before I press the trigger, we're using my mouse. I'm holding back my mouse from pressing the trigger. So when I press the trigger, let me see your answers here. Where do you expect to see the arrow regarding cyclic AMP with pertussis toxin? Increase arrow, decrease, or no change? Increase, decrease, or no change? And the answer is it will increase. Cyclic AMP levels rise with pertussis toxin ADP ribosylating alpha I. All right. Now, how do we explain that? And you explain that by saying, what is alpha I normally supposed to do? And you answer by saying, well, alpha I inhibits adenosylase from making cyclic AMP. That's the normal situation. And now, because the cyclic AMP levels rose, what do you think happened? By putting ADP ribose onto alpha I, what do you think happened to alpha I? There you go. Perfect. What do you think happened to alpha I? That's right. You inhibited the inhibitor. That's right. You inactivated the inhibitor. A double negative. There it is. You inhibited the inhibitor. Double negative. There it is. There it is. And so therefore, by inhibiting the inhibitor, what happens to what happens to adenosylase? Adenosylase says, I'm free. I'm free at last. I can now start making cyclic AMP. And so it starts making cyclic AMP, and levels rise. And then you get all the pertussis situation that goes on in pathology. Again, you know, that's all the downstream situation that goes on. These are all downstream situations. But the biochemical explanation is right there. In all three cases, cyclic AMP levels rose in different tissues, causing the different pathologies here. There it is. Beautiful, beautiful how it works. That's right. Now, before we leave this here. Now, without looking, don't go looking in your book. Hey, I hear somebody turning the page. Is that you, Vio? Vio, are you turning the page? Or is that you, Fumni? Nobody turn the page. No, okay, nobody turning the page. No turning the page. All right. Now, what was the name of the other two toxins that also worked by ADP ribosylation? What were the other two toxins? What were the other two toxins? What were the other two? There we go. Now I'm talking. There we go. What were the other two toxins? And one of them was diphtheria, and the other was, what was the other one? Not sugar. No. And the other one was not anthrax either. And the other one was Pseudomonas. There it is. Pseudomonas toxin. So those ones also worked by ADP ribosylation of a G protein. Now, without looking in the book, what was the name of the G protein? What was the name of the G protein target? Without looking in the book. That's right, and it was elongation factor 2. Now you can look on page 56. If you go back to page 56, you will see diphtheria and Pseudomonas toxin also work by ADP ribosylation of yet another G protein. And this one's called elongation factor 2 because it binds GTP in order to make the ribosome move in the process of translation, protein synthesis here. Now notice here, the high yield topic, high yield, 5 different toxins all work by ADP ribosylation of a specific G protein. And that G protein target is what they're going to ding you on. You better get this straightened out. The integration of biochemistry with microbiology here. Major, major. You got to get this straightened out here. You cannot, you're going to get a toxin on board exams. Out of 322 questions, at least one of them is going to be a toxin, right? You would think. At least one. And so the sooner you understand that, get this straightened out, the better off. Now, somebody mentioned sugar toxin. Saga. What does sugar toxin do? What's the biochemical mechanism of sugar? How does sugar kill me and you? How does sugar work? That's right. And what it does, it cleaves. That's right. It binds to the 50S, excuse me, the 60S subunit, the large subunit in eukaryotes, the 60S subunit. And there it cleaves a ribosomal RNA. Therefore, the ribosome cannot move on the messenger RNA. A different mechanism of killing is sugar toxin, major item for the national boards. Okay, well done. Those of you you're responding beautifully. Beautifully, you're responding. Even if you give the wrong answer, you're beautifully responding. You're participating. You're learning. And therefore, you're my type of person because you don't want to whatever that other M word is. You want to master the material, which is the perfect thing to do. Now, the last thing here is to finish off this chapter, and then we'll take the break. The oncogenic mutations. RAS protein, remember we talked about it earlier, it's a G protein dysfunction. Any of those G proteins, these three-letter things like Herb and Mick and June and so on, these are those three-letter oncogenes that Dr. Barone will talk about. Remember that these are actually proto-oncogenes, which are normal. But if any of them get a mutation in it, like RAS, then cells will start to grow when they should not grow. And notice, the RAS is in cells in G1 phase. In G1 phase, epithelial cells that line the colon and the bladder and so on. And those cells are they're waiting for growth factors now to stimulate the bind to the tyrosine kinase receptor to stimulate gene expression. And so again, that issue of gene expression is now going to be turned on when RAS binds GTP. And in this case here, it's doing it when it should not be because of mutation. Okay, so we're done for this next hour. Wow, time went fast. Man, hard to believe. Okay, so we're done with this chapter, and man, it's amazing how time flies. Okay, so let's take our 10-minute break, and I'm going to go off camera again, and I will be back shortly here. Okay, and more. Wait to the next chapter. Vitamins. You're going to be glad you came tonight. All right, see you shortly. Alrighty, so we're back for the third hour. Time's flying. A lot of good stuff coming your way, and in the next 2 hours, man, I'm telling you right now, there's a lot of high yield stuff. And so the next chapter again, we still got a ways to go before we get to individual pathways. All we're doing, I've been here for 6 days and 2 hours, and all I'm doing is setting the stage for metabolism, because that's what life is about, is metabolism. And we still have a ways to go. We got to talk about vitamins. The previous chapter, we talked about hormones that affect individual metabolic pathways. Before that, we talked about enzymes, which carry out individual metabolic pathways. So we're setting the stage for the big picture of life, and that's those metabolic pathways coming up here. Okay, now this chapter is an extremely high yield chapter, and it's vitamins. Now let me say something about vitamins here to preface this chapter. Of all the stuff you had in the first 2 years of medical school, all that stuff you had, physio, pharm, this and that, of all the stuff you've had, if I had to choose, one of the nice things about being a veteran of Kaplan Medical, 17 years, I've talked to students all over the world who took board exams, blah, blah, many times over, talked to many students, lots of emails and things from students. And if I had to choose the number one topic, the highest of the high yield topics for national boards, it's this chapter, vitamins. I have never, in my entire medical career teaching students, I have never encountered a medical student who didn't have at least one vitamin question on the national board. Never. Maybe there is a medical student out there that never had a vitamin question. I have never encountered that student yet. So what I'm saying is you got to know your vitamins. You must know them because it's the highest of the high yield topics. I cannot guarantee it, but all I can say is you better know it because you're going to see at least one vitamin question on board exams. Now let me prove this to you as an example. Let me prove this to you. About a year ago or so, I was in Chicago doing the live lectures there at the Kaplan Center. And then that was a year ago in February 2013. And then about a year ago at this time, in May, I got an email. I was sitting in my computer and sitting there in my office, and then I heard the ding. You got mail. So I went to my email, and what it was an email from a medical student who took that Chicago course, and this student was named Jeffrey. He was from one of the medical schools in St. Lucia, and this is what he wrote. He said he did well. He took the board exams and he did well. In fact, he did really well. He got at St. Lucia. They told me he got something like 260, something like that. It was outstanding. I mean, it wasn't just a pass. He did really well. He did extremely well. But notice what he wrote. Notice what he wrote in this second paragraph. He wrote that he had 3 vitamin questions because I hammered home what I just told you. You better know your vitamins. You better know your vitamins. And listen to what else he wrote in the second sentence here. Look what he wrote. He said he had 2 pedigree questions. Remember I told you better know your pedigree? And then what else he said? And he had 2 questions related to mitochondrial inheritance. Remember I told you you better know your mitochondrial inheritance? Remember that? Yeah, notice what he's saying here. He had 4 questions. The way I interpret his English is the same as my English. It says 2 plus 2 was, last I heard it was 4. 2 plus 2 is 4. Although you never know about the medical education, about the education they're having in the United States, this Common Core curriculum. The Common Core curriculum, what they do nowadays, they're trying to push it on the states where they say, in a sample question in mathematics, if somebody has 2 oranges, and you give them 2 more oranges, how many dogs live in England? That's their logics, But the idea is here. They're saying 2 plus 2 questions on inheritance. You better know your modes of inheritance. Just what I wrote here. Just what I wrote. And notice what he also wrote here. In another sentence, he wrote, He said he had a question involving a technique to analyze a particular condition. And said the trick was knowing what disease was it, and then ask you, well, how do you find a test? What's the best test for that technique? Here, remember that I gave you an example of this. Remember that question that dealt with beta thalassemia. In beta thalassemia, you had to figure out the patient had beta thalassemia, but then you had to know that the patient had what causes beta thalassemia. It's a splicing defect, and what's the best way to determine splicing defect diseases is the northern blot. So he got a question. He didn't say what it was, but it was similar to that. So it makes me feel good that I'm not wasting your time. And notice what else he wrote in the first sentence. It made me feel real good. It made my day. Look what he wrote. He wrote, encouraging us to push hard further. He was. I mean, when I read that, it made me feel so good. In my own little world, I'm helping people out. You know, that I'm pushing students. I'm pushing them not to memorize. I'm pushing students to learn, to master the material, to want to know the material. You know, push harder, because that's what it's all about. You can do it. You can do it just like Jeffrey. You can do it just like him. I get emails like this all the time. I'm telling you right now, I get emails like this I just as an example of one. I get emails like this all the time from students, and but I'm pushing students because I want them to learn instead of memorizing here. And if you just follow, like when I give my lectures here, as long as you're following me, as long as it makes sense, as long as it's logical, and as long as I'm clear and getting across, I got your back, because I know what's important for board exams. I know what's important for board exams. Nothing to fear. Just follow me. Just follow me. I got your back. I know. As long as you're following me, you're going to do well. And you can have a big sense of relief. Big sense of relief. Oop, back over. Right? Big sense of relief. Right? There you go. There you go. Just a big sense. Yeah. Yeah. That's all you need. That's all you need. All right. Now we go. Got to have some humor with this as well. But the idea is, put that up there tonight in my PowerPoint, because I thought, man, yeah, this student should be relieved. You know, that somebody's watching after them. Somebody got their back. As long as I do my part, as long as you do your part. I keep telling you, you got to do your part. Your part is to pay attention. Don't get distracted. Follow me. Participate. I got your back. You will do well. You will. I have faith in you. I have faith in you. Even if no one else does, I know you can do it. I'm pushing you. I'm going to push you to do it, because I know you can do it. I know you can. No doubt in my mind. You just got to work at it. No substitute. All right. Now what we're going to do is, well, first what we're going to do here is sometimes it's scary. I get it. Sometimes it's scary. I get it. But that's part of life. You know, it's life was not meant to be easy. Life means sometimes you got to work hard. I worked hard to be where I'm at right now. I worked extremely hard to be where I'm at. I've been blessed more than I deserve to be blessed, to be right in front of you right now. Really, honestly, I don't deserve to be here, but I worked at it, and I listened and this and that, and you can do the same thing. There's nothing magic. All the stuff that I'm telling you about regarding biochemistry, there's nothing. I wasn't born with this material. It's just simply I wanted to know it to be able to convey it to the students. You could do the same thing. Nothing magic. You got to work at it. All right. Now what's coming up here is, of the vitamins, there are two classes of vitamins. There are the lipid-soluble hormones and the fat-soluble hormones, and then the water-soluble hormones. Now the lipid fat-soluble hormones, those will be discussed a little bit later. There's 4 of those. But the water-soluble hormones, what I want to do here is not hormones, I keep saying hormones, the vitamins. So the fat-soluble vitamins and the water-soluble vitamins. Now the fat-soluble vitamins will be discussed a little bit later. The water-soluble vitamins we're going to discuss in a nutshell here, in a nutshell. What I want to do is, in this column here on the left, On this column here on the left, what I want to do in blue is outline the difference between the vitamin and the coenzyme, because they're not the same. The vitamin, after you eat it, is made into a coenzyme, and enzymes use coenzymes, they don't use vitamins. Don't worry about the name of the enzymes that use that coenzyme. That's discussed later on in metabolism. And this column here, what I want to do is outline in red what is one word or one phrase in which that vitamin coenzyme is involved in. You know, like for example, tomorrow morning when you eat, you wake up, you go to your kitchen, and you are eating some cereal, and you take those Cocoa Puff cereals, and you look in the margin of the cereal box, and you see all these vitamins like riboflavin, folate. What do the vitamins? What does riboflavin do in one word or one phrase in your body right now? What does that particular vitamin do? That's what I'm going to do here in this column in red. And in green, here on the right side, is highlight is what is the hallmark of the deficiency of that particular vitamin. What do you want to look for on board exams if somebody has riboflavin deficiency? Now, what word or what's the main clinical manifestation? All right, so the first one to talk about is biotin. Now, biotin is a vitamin, and it's used to make the coenzyme called biotinyl lysine. Now, what does that mean? In the active site of three major enzymes, don't worry about them now, we'll talk about them later on, but in the active site of these three enzymes, there is a lysine residue in the active site, and in covalent linkage to the lysine is biotin, so that's considered the coenzyme form, biotinyl lysine in the active site of three major enzymes. Now, what's the function of biotinyl lysine? It's involved in carboxylation reaction, the putting on of CO2 on a substrate to make a product. Notice the three enzymes that require biotin are carboxylases that put CO2 on a substrate to make a product. Now, in terms of the pathologies associated with biotin deficiencies, they are very rare biotin deficiencies, MCC. Most common cause of biotin deficiency is people eating raw eggs almost exclusively. And the reason is because in egg white, there is a protein called avidin. And avidin binds biotin very tightly and prevents the biotin from being absorbed. But most people, when they eat eggs, they heat the eggs, they fry, they boil, they cook the eggs, and so the heat denatures the avidin. So that's why avidin is really not a problem in most people. But people eating raw eggs exclusively for some unknown reason likely will end up with biotin deficiency. Now, what is the hallmark of biotin deficiency? And it's alopecia, hair loss. Now, alopecia could be caused for other reasons. But if you see alopecia on board exams, you say, well, they might be asking me about biotin deficiency. Now, thiamine. Thiamine is a major vitamin for board exams. I would not be too concerned about the B number, but thiamine is the vitamin. Now, a word of caution here. A word of caution is that when you go take the national board exams, because you are human, you're going to be stressed out. And when you're stressed out, you may have a tendency to make silly mistakes by confusing words which sound very similar. Here's a great example of this, where two words sound very similar. And I know you know the difference right now because you're not stressed out. But on board exams, because you're stressed and nervous, you might confuse the word thiamine with the word thiamine. These are not the same. These are not the same. Thiamine on the left side is a vitamin. Thiamine on the right side is a base you find in DNA, and they have nothing to do with each other. But you can imagine on board exams how you might make a silly mistake by confusing these two words with each other. All right, now there's other examples of this coming up later on in metabolism. Some words sound very similar to each other. Oh yeah, I know the difference. That's because you're not stressed out here. but we'll talk about those other examples later on. Now, the coenzyme form of thymine is thymine pyrophosphate, TPP. TPP. Don't confuse it with TTP. TTP is a nucleotide used to make DNA. TPP is a coenzyme. Again, don't get these confused. TPP is a coenzyme. TTP is a nucleotide used to make DNA. In the body, there are several enzymes that need thymine pyrophosphate to work, TPP. Don't worry about them now. They will be discussed, hammered home later on in metabolism. Now, what's the function of thymine pyrophosphate? It's involved in decarboxylation reactions, the removal of CO2 from a substrate to make a product. And now, why is thymine such a big deal for board exams? Because there's a lot of pathology associated with it. It's fairly common, and thymine deficiency is well known, and it gives rise to beriberi. And there are two kinds of beriberi. That's wet beriberi and dry beriberi. Now it's easy for me to remember which of the two. I'm going to simplify it here. What's the difference between dry beriberi and wet beriberi? And the easy way to remember them is that dry beriberi is neurological beriberi. Something you have neurological problems, whereas wet beriberi is a cardiac problem. The high cardiac output, cardiac failure, for example. So they have other problems as well. But the easiest way to remember dry versus wet. Dry is neurological, wet is cardiac. Here. Now, of the two, which one comes first? So if you're thymine deficient, which of the two comes first, dry or wet? Which one comes first? Which one comes first? Not the wet, no, it's the dry. Dry is the earlier one. The neurological problems come first. Neurological problems, remember the polyneuritis comes first, all kind of stuff. And so the idea is polyneuritis will come up first in thymine deficiency. And if it manifests to the wet beriberi, then it is a very serious problem. The cardiomyopathy and so on, the dilated cardiomyopathy, that is a very severe, dangerous situation that can go on. The high cardiac output, major problem can go on there. Now, don't worry about it now, but we'll talk about it later on, is that Wernicke-Korsakoff syndrome. Don't get that confused with beriberi. It's not the same. Wernicke-Korsakoff syndrome also is associated with thymine deficiency, but it's not the same as beriberi. Now, what happens here, don't worry about it now, but we'll talk about it later. But in Wernicke-Korsakoff, what happens there is an enzyme that's not going to work. And when that enzyme doesn't work because of thymine issues, then it gives rise to the Wernicke-Korsakoff syndrome. And that enzyme is called transketolase. Don't worry about it now, but transketolase is in the hexose monophosphate shunt. Once we get to the hexose monophosphate shunt, that's when we'll review the difference between beriberi, Wernicke-Korsakoff, alcoholics. Why do alcoholics present with Wernicke-Korsakoff? At that point is where we'll talk about the issue of these, how you distinguish these different kinds of clinical manifestations. They're not the same. Now sometimes people ask me about ophthalmaloplasia. What does that mean in Wernicke? Ophthalmaloplasia, that's just where, if you have here's a female who has Wernicke-Korsakoff, thymine deficiency, and the idea is that if I shine a beam of light across her eyes, she has a characteristic wide-eyed appearance, and if I shine a beam of light across her eyes will not be able to follow the beam of light. That's what this word here means, ophthalmaloplasia in Wernicke. Okay, let's move on. Come back to thymine, major vitamin for boards. Another major vitamin is niacin. Now, niacin is used to make the coenzymes either NAD or NADP. Now, let me stress, these are not the same. NAD is a different coenzyme than is NADP. They are not the same. They are not interchangeable. There are many enzymes that need either NAD or NADP, and they're all classified as dehydrogenases, and there are many dehydrogenases in the body. For example, lactate dehydrogenase, alcohol dehydrogenase, glucose 6-phosphate dehydrogenase. And these dehydrogenases either use NAD and other ones use NADP, and they're not interchangeable. Now, now, if you had to ask, what's the function of NAD or NADP, and with these dehydrogenases as a surname, and you answer they're involved in oxidation reduction reactions, the movement of electrons from one molecule to another. I know medical students hate the term oxidation reduction, but again, that's part of life. It's part of life. Oxidation reduction. The removal of electrons and the putting back of electrons into carbon. That's what life is. Stay tuned, stay tuned. It's all coming up here. Okay, now moving on. Now the next thing here is the issue of niacin deficiencies, also well known, and that's called pellagra, characterized by the 4 Ds. Diarrhea, dementia, dermatitis, and the biggest D of them all, death. Now, the issue of dermatitis. Here's a woman who has pellagra. Look at that dermatitis. It is very severe dermatitis. It's not some little rash on the hand. This is severe dermatitis that goes on in pellagra, in the hands as well as around the neck. So again, major item is this issue of pellagra. Now, another item that's high yield for boards is you must know that we talked about it on Saturday, that tryptophan is used to make niacin. Don't worry about the biochemistry, how you convert tryptophan to niacin. That's not important. But you should know that with some tryptophan that you eat, some of it is converted to niacin. And it turns out it's not enough niacin. Therefore, you have to get more of it from the diet. All right, now here comes a very important item here, because this was on the boards last summer. Here it comes. Now, what's the name of that? You probably talked about it in physiology, but you will pathology next week as well with Dr. Barone. But what's the name of that genetic disorder in pathology in which the transporter for tryptophan is deficient in the mucosa, such that you do not absorb very much tryptophan, and therefore the body levels of tryptophan are low, and the same transporter is defective in the kidney, and so therefore you do not reabsorb what little tryptophan you have. So you pee it out into the urine, so the body levels of tryptophan are abnormally low, and the patient ends up with pellagra-like symptoms. What's the name of that disorder? That's right, that's called, well done everybody, that's called Hartnup disease. Now I want to stress, they don't have pellagra. Pellagra is a nutritional disorder. They have pellagra-like symptoms. That's the diarrhea, dementia, and the dermatitis. Please understand, that pellagra is by definition a nutritional disorder. Hartnup is a genetic disorder. They're not the same. They're related, yes, to each other, but they are not the same disorders here. One's genetic, one's nutritional. So please get that straightened out here. Okay, now so again, major. That was on the board exam last summer. That was on the last summer's exam. Hartnup disease, major. I got your back. I got your back, as long as you're paying attention. Now, of the water-soluble vitamins, one of the top 2 for the national boards, major one, you got to know, if you think thiamine is important and niacin, here's one of the top 2 water-soluble vitamins for boards is folic acid, also called folate, used to make tetrahydrofolate as the product. Now, now there are a number of enzymes that need tetrahydrofolate, and they're in pyrimidine purine metabolism. Yeah, so we'll talk about that issue on Friday. But the idea is, now what's the function of tetrahydrofolate? And the answer is it's involved as a 1-carbon carrier. In other words, THF is a carrier of one carbon, and use that one carbon to put it onto a substrate to make a product. Now see this little box here, this little box. That little box why is folic acid so high yield? Is because this little box right here, this little rectangle, is the target of so much pharmacology, that one carbon transfer from the THF to put it onto a substrate to make a product. As an example, let me ask you this. What's the difference between uracil and thymine? Thymine on the right side here. See that thymine on the right? What's the difference between uracil and thymine? What's the difference? What's the difference between uracil? That should be immediate. That should be very fast. That's right. It's a methyl group. That's right. A methyl group. If you take a methyl group, you put it on uracil, you form thymine. And so therefore, where did that one methyl group come from to put it on uracil to form thymine? It's coming from a THF carrier, tetrahydrofolate carrier. It's carrying that one carbon, that methyl carbon. You put it on uracil to form thymine. And that is the target of so many drugs like methotrexate, anti-tumor drugs, antimicrobial drugs like sulfa drugs, anti-parasitic drugs coming up later on. But many drugs target that one carbon transfer from THF onto a substrate to make a product. Major item. Now that's pharm. Here comes on the right side, here's the pathology. Now, folic acid deficiencies are well known. And now at the bottom of this rectangle here, notice here that in early pregnancy, that folic acid needs to be present throughout pregnancy, especially within the first few weeks of pregnancy, because that's when the neural tubes are being developed that Dr. White talked about in anatomy. That's when the neural tubes are being developed, and folic acid must be present. And if it's not present, then the neural tubes do not develop properly, and you can end up with a tragic spina bifida and so on, neural tube defects. Tragic disorder here. Sometimes even before a woman knows she's pregnant, it might be too late. So it has to be present throughout pregnancy, especially within the first few weeks of pregnancy. Now in adults, folic acid deficiencies are also well known in alcoholics as well as a pregnant woman. But in terms of the clinical manifestations of folic acid deficiency, in my opinion, the #1 hallmark is this: megaloblastic anemia. If you see that on the national board, they tell you a patient has megaloblastic anemia, immediately you start saying to yourself, they might be asking me about folic acid deficiency. Now at the top of this, in the first panel here, these are normal bone marrow aspiration. These are mostly erythrocytes, and in the blue here, these are nucleated red cells. And at the bottom of the figure here, these are the megaloblastic nucleated red cells. Look how massive they look compared to normal. Now, this is what goes on in folic acid deficiency. I'm hoping that you already know that. I'm hoping you're sitting back there saying, man, I can't wait for Dr. T to ask me why. In folic acid deficiency, why the red cells look huge. Now, if you don't know, don't answer right now, but I'm not looking for the answer. But I'm hoping you're saying something, man, I can't wait for him to ask me because I know the answer. I want to type it in the public chat pod. But I'm hoping, don't answer. I didn't ask for the answer. I didn't ask for it yet. Don't answer. It's a rhetorical question. It's coming up later. I'm trying to get students to think about it because that's going to be covered later on. This is just a preview, a nutshell of things coming down the road later on in metabolism. But the idea is I'm hoping you're thinking, I know why. No folic acid, why the red cells are huge. That's coming up later. All right, so why the red cell? Natalie, what's the question? Megaloblastic anemia and folic acid deficiency, why? but the answer will come later. All right, now if folic acid is one of the two most important vitamins for the boards, in my opinion, what's the other one? And the other one is B12. You got to know B12. Out of 322 questions, at least one of them is going to be a B12 question here. And now B12 is also called cyanocobalamin. And cyanocobalamin is made by the bacteria flora. So technically you don't have to get B12 from the diet. So as long as you have an active bacteria flora in your colon, they're going to make B12 for you, and then you absorb the B12 by the intrinsic factor. After the absorption, the cyano group is enzymatically removed and replaced with methyl or adenosine, specifically deoxyadenosine, to form the coenzyme form of B12 here. So again, the cyano group is removed and enzymatically replaced. This is not trivia. Later on this week, I'm going to ask you about the pathology associated with this, where some people cannot convert the vitamin B12 to the coenzyme form of B12. Okay, don't worry about it now. In the body right now, there are two major enzymes. Don't worry about them now. They're going to be covered later on in metabolism. Mega enzymes. Only two enzymes in the body require the coenzyme form of B12. Don't worry about them now. But in terms of the function of the coenzyme form of B12, it's involved in something called the vomit pathway. But again, don't worry about what that means. Hopefully you already know what it means, but if you don't know, stay tuned. It's coming up later on in metabolism. Now, why is B12 such a big deal? And because there's a lot of pathology with B12. The pathology usually rises in elderly individuals, people who are aged individuals, is where you're likely to see B12 problems, and all the characteristics of B12 deficiency. Besides the progressive peripheral neuropathy, the number one hallmark, there it is again, megaloblastic anemia. Now, I'm not going to show you that previous figure, but remember those megaloblastic cells? I'm hoping, don't answer yet. That's coming up later. But I'm hoping you're saying to yourself, man, I can't wait for Dr. T to ask me, why is it in B12 deficiency the red cells look huge? Because I'm telling you right now, the National Board loves that. I hear that multiple times a year from medical students. They ask a question on board exams, they give you the clues that the patient has megaloblastic anemia, and they're going to want to know, how do you know? Is it due to folate deficiency or is it due to B12 deficiency? It's all coming up later, all coming up. Hopefully you're here the rest of the week. Mega stuff coming your way. Now, in addition, they have progressive peripheral neuropathy, the tingling of the fingers and so on. Why is that happening? Why? All coming up later. All right, now moving on. Yet another major vitamin for board exams. Did I say the vitamins are important for boards? Hopefully I did. Hopefully I didn't forget to tell you got to know your vitamins. Next one here is B6, also called pyridoxine, and it used to make the coenzyme called pyridoxal phosphate, the coenzyme form of B6. Now, one thing I want to say about B6 is the pyridoxal phosphate. Notice the suffix here, the AL ending, pyridoxal phosphate. That's telling you something. AL in organic chemistry is telling you it's an aldehyde group is important. If you see an OL ending, a hydroxyl group is important. You're going to see this later on in the next hour, like with something called retinol and retinal. Retinol, the hydroxyl group, is a different molecule, has a hydroxyl group than retinal, which has an aldehyde group. So here, pyridoxal, AL ending, is telling you something. There's an aldehyde group in the molecule. And what you need to know about aldehyde groups is that in metabolism, aldehyde groups react with amino groups very tightly. So aldehyde groups react with amino groups. Now, if you're thinking, well, so what? Okay, that's more chemistry. I can't stand chemistry. Who cares that aldehyde groups react with amino groups? You should care because because this has everything to do. You must know that aldehyde groups react with amino groups because that has everything to do with something called hemoglobin A1C. If you want to know what hemoglobin A1C is coming up later on, you got to know that aldehyde groups of glucose react with amino groups of hemoglobin, forming hemoglobin A1C, also called glycosylated hemoglobin, also called glucosylated hemoglobin. It's all coming up a little bit later. You'll never understand that stuff unless you know that aldehyde groups react with amino groups. There it is, all coming up later. Now what's the function of pyridoxal phosphate in the body? You would say, well, because the aldehyde groups react with amino groups, you would say, well, it's got to be involved in transamination, the movement of amino groups from one molecule to another. A lot to say about that on Friday. That you need to know that aldehyde groups react with amino groups here. Okay. Now in this rectangle right here, major rectangle, B6 is such a high yield vitamin because in this rectangle right here are 4 disciplines: pharm, micro, pathology, and of course biochemistry. Biochemistry is everywhere. So 4 disciplines in this one little measly box here. So here it is, isoniazid, everybody knows. Let me ask you this, for what reason, and since you had micro, for what reason is isoniazid used to treat what disease? Everybody knows this. That's right, and it's tuberculosis. And why you end up with B6 deficiency? Because isoniazid binds B6 and prevents B6 absorption. And it also interferes with B6 action. So again, that's why they end up with B6 deficiency with isoniazid therapy here. Now they also have sideroblastic. Now in terms of the pathology, the hallmark, they have sideroblastic anemia, chylosis, convulsions, and so on. But in my opinion, the number one hallmark of B6 deficiency is not even shown here. The number one hallmark of B6 deficiency. Let me see your answer. From pathology, what is the number one hallmark of B6 deficiency? Neuropathy, and that's not a hallmark. B12 deficiency gives you progressive peripheral neuropathy. A lot of things cause vomiting. A lot of things cause vomiting. I mean, the hallmark. In other words, what do you want to think of? The hallmark being, if you see this symptom alone, this clinical manifestation alone, it's pointing you in the right direction. A lot of things cause vomiting. Okay, which anemia? We've got a lot of anemias out there. What do the red cells look like in B6 deficiency? And Takir, there it is. That's right. And the answer is microcytic anemia, where the red cells look tiny, in contrast to megaloblastic anemia with B12 and folate deficiency. In B6 deficiency, the red cells look tiny. What else do they look like? Let me see the answer. What else do they look like besides being tiny? That's right. Give everybody a chance to write. That's right. Leo and Gisela. That's right. And that's right. And they look hypochromic. That's right. Maritza. That's right. They look hypochromic. They have no color. So they're microcytic hypochromic cells. Hallmark of B6 deficiency. Now the idea here is now you should also ask the why question. Why is it that in all these, many, not all, but many of these vitamin deficiencies, it's the red cells that are suffering? You should ask. Do you ever ask in pathology with all these vitamin deficiencies? It seems like the red cells are suffering. Megaloblastic, microcytic cells, and the reason is because nucleated red cells are cells in G1 phase, and so those cells need to replicate DNA and grow, eventually differentiate, and they need a lot of vitamins, different kinds of vitamins that make things and things along that line. If you're missing one of the vitamins, it's the red cells primarily are affected. But in addition, any cell in G1 phase will be affected. That's why you have GI tract problems with some of these vitamin deficiencies, and skin cell problems, dermatitis, because those cells are in G1 phase. Any cell in G1 phase needs vitamins to replicate DNA, to grow, to divide, to make proteins. So that's why they suffer. That's why you usually don't have problems like liver problems with vitamin deficiencies. Usually it's the cells in G1 phase. All right, now the question why? Why is the other question why? Why is it that in B6 deficiency the red cells are tiny and they have no color? And the answer is simple. This is where you could memorize this fact: microcytic hyperchromic cells, and sideroblastic cells as well. But if you think about it, it makes sense. The why question. And the way you explain this by saying, again, I'm a simple guy. I ask simple questions. Why my wife calls me a simpleton. Is that a good thing? Call me a simpleton? I don't know. She calls me a simpleton. I take it as a good thing. But in any case, the idea is I'm a simple guy. So here's a simple question. To try to understand why is it in B6 deficiency they have no color and they're tiny, why? Well, you would say to yourself, well, why are red cells red? Let me see your answers here. Why are red cells red? Let me see your answer here. Why are red cells? Simple question. Why are red cells red? If somebody asks you, why are red cells red? What would you say? That's because of the heme, not the globin. It's the heme. Heme is a red-purple pigment. And so here it is. You would say, therefore, if heme is the cause of the red in red blood cells, then when you don't have B6, then that must mean there got to be and you have no color, that must mean that in heme synthesis, there got to be a major enzyme that requires B6. No B6, the major enzyme in heme synthesis will not work. Therefore, you don't make protoporphyrin. Therefore, no heme. Therefore, no color. And therefore, the red cells don't develop, and they are microcytic and no color. There it is. The top enzyme, right? Don't worry about it now. Coming up later, ALA synthase. The top enzyme of heme synthesis requires B6. Without it, you don't make protoporphyrin. All right, now here it is. Another item, just how it makes sense. Making sense out of things. What does sideroblastic anemia mean? What does that mean? Sideroblastic anemia. What does that mean? Sideroblastic anemia. Let me see your answers here. That's right. What it is. Here's how you explain. Here's how you explain this. As long as you know a little bit about biochemistry, we'll talk about later on this week, that you already should know since you had the course in the first year of medical school. But here it is. In heme synthesis, the last step of protoporphyrin synthesis occurs in the mitochondria of the nucleated red cells, in the mitochondria. So you make protoporphyrin in the mitochondria in the last step, and in the last step of heme synthesis, in the mitochondria, you have an enzyme that puts iron in a +2 state into the protoporphyrin to make heme. So the last step is in the mitochondria, where you take iron, you put it in the protoporphyrin to make heme. Now, if you don't make protoporphyrin, because you don't have B6, you got to ask, what happens to the iron in the mitochondria? Well, the iron in the mitochondria stays in the mitochondria. And when it stays in the mitochondria, that is sideroblastic. That's what it means. It's that there's too much iron in the mitochondria. It forms like a ring around the nucleus. These are mitochondria. They go to form a ring around the nucleus. It is simply too much iron in the mitochondria. They're trapped in the mitochondria. That's what sideroblastic means. All because of something wrong with the biochemistry. There it is. All right, let me finish this up on this page, then we're done. Then we'll take the break. Okay, major vitamin B6. Moving on, riboflavin used to make FAD. FAD is the coenzyme, and it says here there are many. In fact, there are just 3 enzymes. They're all classified as dehydrogenases that use FAD. And what's the function of FAD? It's involved in oxidation reduction reactions. It's the movement of electrons from a substrate to make a product. Now, of the clinical manifestations of riboflavin deficiencies, in my opinion, there it is right here, chylosis and stomatitis. The chylosis, everybody knows. If I ask you what chylosis means, you would also you'd be pointing to your mouth, and that is chylosis. They also have a characteristic cracked tongue in riboflavin deficiency, and they might also have a magenta colored tongue. Magenta is like a pink purple color there. Magenta colored tongue would be also characteristic of riboflavin deficiency. Major issue for boards is the characteristics of riboflavin deficiency here. All right, now moving on. Now, ascorbic acid, crusty lip, they can also have that characteristic as well. Now, ascorbic acid. Now, ascorbic acid, if I were to or vitamin C, if I were to ask you, what is the coenzyme form of vitamin C? And you don't use your keyboard because that's a trick question. There is no, vitamin C is used as is. It's the only water-soluble vitamin that's not made into a coenzyme. And now there are a number of enzymes, a few of them shown here, that use vitamin C. And so these enzymes are involved in collagen synthesis, catecholamine synthesis, and so on. But another characteristic, function of vitamin C in the body is it's involved as an antioxidant. And later on, once we get to vitamin E, because vitamin E also is an antioxidant, that's where we'll review why do you need antioxidants in the first place. What's all this issue of free radicals all about? All coming up once we get to vitamin E. Now, the issue of scurvy. Now, we talked about this earlier in collagen, that scurvy has 2 hallmarks. One of the hallmarks of scurvy is the elastic skin. That's already been reviewed, and by now you should be able to verbalize why is it in vitamin C deficiency. The skin is elastic. What we have not yet reviewed, and we will in the next hour, is this. Why is it in vitamin C deficiency? Why do they bleed? Why do they bleed? Why do they have bleeding gums and all that in vitamin C deficiency? Now, I'm hoping you already know the answer. But if you don't know why, stay tuned, because we'll talk about that in vitamin K deficiency. Because in both vitamin K deficiency, vitamin C deficiency, they bleed. And we'll do the biochemistry explanations coming up in the next hour. And the National Board loves it, because they're going to want to know if somebody's bleeding, how do you know? Is it due to vitamin C deficiency, or is it due to vitamin K deficiency? And you'll never understand it unless you know the biochemistry first, that we'll talk about after the break. Okay, one minor thing, last thing here, is pantothenic acid. Now, pantothenic acid is the least important vitamin for the boards. It's used to make coenzyme A. There are many enzymes in the body that need coenzyme A. And so if you had to say, what's the function of coenzyme A? It's involved as a carrier of fatty acids, but not just fatty acids. Used to carry acetate, used to carry HMG, those type of things. But why is pantothenic acid not important for boards? Because pantothenic acid deficiencies are extremely rare, if non-existent here. So that's why they don't worry about it for the boards. But although pantothenic acid is essential for life, it's essential for life. But for the boards, it's not important. And the reason why, because as long as you're eating something, your pantothenic acid, basically, it's in that food. So at the break coming up here, is if you get hungry, all you got to do is lick the floor that you're standing on. And you'll probably get enough pantothenic acid that way. So again, pantothenic acid is basically in all the foods that you eat. Okay, what we're going to do now is take a 10-minute break and then come back and then get into the fat-soluble vitamins in more detail. The fat-soluble vitamins, the first one being vitamin D. Okay, let's take a 10-minute break and see you in 10. Everybody, welcome back for this last hour. So we are done with the water-soluble vitamins. A lot more to say about them in metabolism. And what's coming up next are the 4 fat-soluble vitamins. And the first of the 4 to go into some detail is vitamin D. Okay, now you're likely to see something about vitamin D on board exams, and you probably already talked about it in physiology and endocrinology portion, and you're talking about it now, and you're probably going to talk about it again with Dr. Barone because of the pathologies as well. Speaking of the pathologies of vitamin D, where you see vitamin D deficiencies in children, it gives rise to rickets, and in adults, gives rise to osteomalacia. Now, I didn't ask because I knew you knew this. I mean, every medical student knows this, rickets and osteomalacia here. Okay, now in terms of the biochemistry and the physiology and the anatomy, you got to know location, 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 where things are happening. Absolutely essential. Here, you got to know this anatomy coming up here. But even though it's called a vitamin, vitamin D, technically it's not a vitamin because the body can make it. You can make vitamin D. You don't have to get it from the diet. And what happens there is in cholesterol synthesis. Now we'll talk about cholesterol synthesis later on this week. Next to the last step in cholesterol synthesis, there is a molecule that can be used to make cholesterol, but a little bit of that molecule called 7-dehydrocholesterol can be diverted out of cholesterol synthesis. You don't need very much. So a little bit of the 7-dehydrocholesterol, instead of making cholesterol, can be diverted out of cholesterol synthesis and be used to make vitamin D in vitamin D synthesis here. So that's why it's not essential, because you can make cholesterol and therefore you can make vitamin D. All right, now in terms of the biochemistry, what happens to it? Now the name of the enzyme that makes it, not important. But once 7-dehydrocholesterol is made and it goes to the skin, and then what happens is a photon of UV radiation will cleave the B ring and then opening up the B ring and thereby making cholecalciferol, also called vitamin D3, as the product. So 7-dehydrocholesterol is considered the provitamin used to make the vitamin called vitamin D3. Okay, now a couple comments about this this arrow here. I gotta pause and talk about this arrow. That's going on this reaction is going on in the skin. Now let's talk about cholesterol a minute, this arrow. How much light do you, by the way, do you know the structure of cholesterol? I mean, no one expects you to know the structure of cholesterol. But in your mind, can you picture roughly what it looks like? Not all the gory details, but I mean, we talked about it earlier. You should know roughly what purines look like, what pyrimidines look like, what the amino acids look like. You should know roughly what they look like without the details. Same thing here with cholesterol. You should know roughly what cholesterol looks like without the exact details. And hopefully you're picturing in your mind 4 rings with all these appendages. That's cholesterol. 4 rings with all these appendages. And it turns out, and this is crucial for you to understand, it turns out that you and I have enzymes to put the 4 rings together. But we have no enzyme capable of cleaving any of the 4 rings of cholesterol. We have enzymes to add to the rings, to subtract from the rings, but no enzyme capable of cleaving the 4 rings of cholesterol. It's an amazing thing, cholesterol. It's one of the very few substances, once you make it and have it in your system, in the body, you have no biochemical way to get rid of it. This is unlike the other macromolecules in the body. Like the other macromolecules like carbohydrates, fatty acids, and amino acids. When you want to get rid of them, you can burn them to CO2 and water. But cholesterol, you cannot burn it to CO2 and water. You can take the cholesterol. Why? Because you cannot cleave the cholesterol rings. You can add to the rings, subtract from the rings, but you can't cleave the cholesterol rings. Cholesterol is not a fuel. You cannot burn it to CO2 and water. You have no enzyme capable of cleaving the 4 rings of cholesterol. That's why, by the way, that's why cholesterol is such a problem for human beings. Once you have it in your system, you and I are going to have a hard time getting rid of it because it can't burn it to CO2 and water. This is the only arrow in biochemistry in which one of the rings of cholesterol is cleaved. And notice, it's not an enzyme that does this. It's a photon of UV radiation. Remember, a photon is an electron. And a photon of UV radiation will cleave the B ring, generating cholecalciferol as the product. The only place in biochemistry where one of the rings of cholesterol is cleaved, non-enzymatically. Now, how much light do you need? Now, it's been estimated that you and I need about 15 minutes of exposure of your cheeks every day. 15 minutes of exposure of your cheeks. And by the way, that's the cheeks on your face. 15 minutes of exposure of your cheeks. You know, like if you go down to Rio de Janeiro, a beautiful city. If you go down to Rio de Janeiro, there's other cheeks that are being exposed. But in any case, I'm talking about the cheeks on your face. Go to Copacabana Beach, you know, Ipanema. Ooh, the other cheeks are being shown here. But 15 minutes of exposure for cheeks, not very much. Now you would think, 15 minutes of exposure for cheeks, what's the big deal? It is a big deal in certain parts of the world. For example, if you ever go to the Arctic Circle in January or February, next January or February, and why? Because you're going up there to look for Santa Claus. Because Santa Claus didn't give you the gifts you deserve. So you go up there looking for him, hunting him down, looking for him. And now if you go up in January, you go there in January and February, there is no sunlight. And those people up there in the Arctic Circle, they are prone to vitamin D deficiency. But as long as they're eating saltwater fish or egg yolks or whatever, plenty of vitamin D that way. In other parts of the world, for example, in Saudi Arabia. I've been to Saudi Arabia the last 4 years. I think I might have mentioned I'm going back again in Jeddah end of next month. And when I go to Saudi Arabia, because of the culture, as you know, a lot of not the religion, it's because of the culture. A lot of the women are wearing hijabs and wear the black outfit where the only thing that's being exposed is their eyes. And when I talk to the female medical students in class, they're wearing the hijabs and so on. And when we talk about this very thing, they tell me they're well aware of it. They're well aware of the issue of vitamin D, and they take vitamin D supplements, those type of things. And even surprisingly, even the male medical students tell me that they in Saudi Arabia, they tell me that they can be exposed. They are prone to vitamin D deficiency, even though they're dressed up in Western garb. And so I was talking to the medical students there. I said, well, why is that? I mean, why are the men exposed? Why are the men prone to vitamin D deficiency? And the students laugh at me and they say, did you go outside today? It was like July. I said, did you go outside today? I said, yeah. And they said, that's why, because nobody stays outside. In Riyadh, where I was in Riyadh last summer, and I think I was there in June or July, I can't remember. But it was so hot, nobody stays outside, you know. And but I was able to go to the store, by the way, in Riyadh, and I got a nice T-shirt. I'm wearing it right now. T-shirt I got in Riyadh. This one I show you. This T-shirt I got. See this T-shirt I got? You can't see it. We can't see it at the front. I'm showing you the back. See the back there? The back. See that? Riyadh there. Riyadh. There it is, man. I got a nice Harley shirt in Riyadh, along with my collection of shirts. But in any case, I was able to stop there. But even there, going to the store, it was so hot. And in fact, not only that, last summer I was also in Dubai. And it was hot there as well. It just, people don't stay outside. They just don't stay outside in July and February, and they are prone to vitamin D deficiency here. So again, it's a cultural issue in Saudi Arabia here. Okay, so again, now coming back to this issue here. So so all you need is about 15 minutes of exposure to get the vitamin D3. So then what happens is, then the vitamin D3 then heads over to the liver, and there is an enzyme called 25-hydroxylase. The name tells you what it does. It puts a hydrox group on carbon 25, making 25-hydroxycholecalciferol as the product. And what you need to know about that product is it is the storage form of vitamin D. The liver makes that vitamin D a little bit stored in the liver, but most of it is stored in the adipocytes. And you have about a month's supply of vitamin D in the adipose tissue. All right, so then when do you need that vitamin D? Here comes the integration with physiology. Now, calcium. Now, everything I'm going to talk about here is calcium, but phosphate's also a major player here. But the idea is, now calcium is one of the very few substances in the bloodstream the body cares how much is there, just like glucose we talked about earlier. But calcium is when the blood calcium is too high or too low, the body sends out hormones to fix the problem. So here it is, that when blood calcium levels drop below 2.5 millimolar, a number you don't have to know, but if it drops below 2.5 millimolar, that's hypocalcemia. And then that stimulates the parathyroid gland to secrete parathyroid hormone. And PTH has two target organs. Now, it's all logical without me telling you. If you were told that PTH has two target organs, what do you think, what makes sense to you are going to be target organs to try to maintain blood calcium levels? What's one target organ going to be, and what do you think it's going to tell it to do? Okay, one of them is going to be the bone, right? That's logical sense. And what do you think it's going to tell the bone to do? Increase calcium removal. So it increases calcium removal by indirectly activating the osteoclasts there. Again, that's all talked about in physiology with Rankle and all that. But the idea is you're removing calcium from in the bone here. Now, in addition, the other target organ is the kidney. And what it's going to tell the kidney to do is increase calcium reabsorption. Don't pee it out. Put it back into the blood. Just makes sense. Now, in the kidney, the other target function of PTH is to stimulate a medium yield enzyme called 1-alpha hydroxylase, stimulated by PTH in the kidney. So it's a renal enzyme. And what's the enzyme going to do? The name of the enzyme tells you. It puts a hydrox group on carbon 1 of 25-hydroxycholecalciferol, making 1,25-dihydroxycholecalciferol, also called calcitriol. And what's important about the product is it's now a hormone, a lipid hormone. And this hormone, like any hormone, has a target organ. And this has two target organs. One of them is going to be the intestine. And in the intestine, it says it increases calcium uptake from the intestine. Now those are 5 words. But what's the biochemistry behind that? And you already know. There's no need for me to go to the details there, because you already know, because you're a thinker. If you were asked, verbalize why it increases calcium uptake in the intestine, and this is the way you would answer. You would say because calcitriol is a lipid hormone, it goes right through the membrane of the mucosal cell. Comes into the cytoplasm and looks for the vitamin D receptor, which is a zinc finger class of receptor, which is a specific transcription factor, which now becomes activated in the presence of the hormone. It then goes in the nucleus and then binds to an enhancer region for a very important transporter called calbindin that Dr. Wilson talked about. And then RNA polymerase comes in, turns on the gene for calbindin. The protein is then the RNA is being made. The RNA is then translated into calbindin protein. and then the transporter is put onto the surface, and therefore more transporter, therefore increased velocity, Vmax, remember all that? You increase velocity, and as a result, more calcium is transported per unit time. Increase Vmax. It makes sense without the detail, because you already know a little bit about chemistry, how it went a long way. There it is. There it is. You should be able to verbalize that. There's nothing magical with me. You should be able to do this as well. And it should make sense to you. Now, the other target organ is the bone. Now, in the bone, what happens there is that in conjunction with PTH, then again, the bone is going to be demineralized. What's going to mobilize? What's going to happen is calcium will come off the bone in the presence of PTH. So PTH and calcitriol together in the bone. PTH and calcitriol together, they work in harmony, they work in synergy. They work together to demineralize the bone. As a result, calcium levels in the blood, because of all the concerted action of PTH and calcitriol together, calcitriol and PTH in the bone will demineralize, and then calcium levels in the blood will start to rise. Here, there it is, will start to rise to try to maintain blood calcium levels here. Now, I want to stress, one of the things the National Boards often does is ask you, well, if PTH is affecting the bone and so is calcitriol affecting the bone, then why do you need calcitriol? And the main reason is because, what's the main difference between PTH and calcitriol? And the answer is it's the intestine, because PTH has no effect in the intestine. Calcitriol does. So again, that's the main organ difference between PTH and calcitriol is the intestine here. All right, now this next part here can be a little dilemma that you need to be paying attention here. A little dilemma. And the dilemma here is that people, when you become an old timer like me, a lot of you are going to have problems with osteoporosis. And so what are you told to do? Like my wife has osteoporosis. Her mother died of osteoporosis many years ago. So what are they told to do? They're told to go to the store and buy calcium pills, calcium carbonate, calcium citrate, supplemented with vitamin D. So how do you explain that if vitamin D, calcitriol specifically, is causing calcium to come off the bone? I mean, it doesn't make sense, right? So how do you explain this? And you explain this by saying, by taking calcium pills, you want to be on the high end of normal. Not that you want to be hypercalcemic, but you want to be on the high end of normal. And by being on the high end of normal, parathyroid gland will not secrete PTH. You don't want PTH to be secreted because you'll be on the high end of normal by taking calcium pills, which is great. Okay. Now, but then what happens is calcitriol, what little calcitriol is around, surprisingly, will now activate the osteoblasts to mineralize the bone. There it is, mineralize the bone. So it starts mineralizing the bone in the absence of PTH. So one more time, calcitriol can either mineralize or mobilize. Put calcium into the bone or take it off the bone. It all depends on the presence or absence of PTH. So that's why. So without PTH around, then the calcium will go into the bone here by activating the osteoblasts. That's why you take calcium pills supplemented with vitamin D. It takes months, if not years. We're talking years. Like in my wife's situation, she's gone from osteoporosis after 5 or 6 years of these calcium pills on. She's gone from osteoporosis to osteopenia. Still got a ways to go. This is taking a long, long time to put the calcium back into the bone here. Now let me ask you this. When calcium levels in the blood rise, What other hormone is being secreted? Let me see your answer. What other hormone is being secreted that Dr. Wilson likely talked about? What other hormone? What other hormone? Calcitonin, that's right. And what does calcitonin do? What does it do specifically? Be careful with the wording. What does calcitonin do? Regulate, that doesn't mean anything. Osteoclast, PTP, and magnesium. Put calcium back in the bone. No, not Lucas. No, good try, but no. And the answer is calcitonin will not put calcium back into the bone. Why calcitonin is good is when calcium is in the bone, calcitonin will prevent the osteoclasts from demineralizing. So it has nothing to do with putting calcium into the bone. It's just preventing calcium from coming off the bone. It's preventing the demineralization process by inhibiting the osteoclasts there. That's what it does there. That's the main, it has other functions, but that's the main function. So that's why you take calcium pills, supplemented with vitamin D in osteoporosis. Calcium will go into the bone slowly by the action of vitamin D, specifically calcitriol. Calcitonin is going to be released to prevent the demineralization by the osteoclast. So that's a slow process, but it works here. Now here comes the pharmacology. Now in pharmacology, there's a new class of drugs over the last 5 to 10 years been on the market, and these drugs are the bisphosphonates to treat osteoporosis. And biochemically, what the bisphosphonates do is like Actonel, Boniva, drugs like that. What they do is they bind to the hydroxyapatite. They chemically bind to the hydroxyapatite, and which has a lot of calcium present, and it prevents the demineralization by the osteoclast. You can envision it like forming like a layer, a protective layer. I like to sometimes view it like a Teflon layer, so that the calcium can't be demineralized by the osteoclast there. So that's how the bisphosphonates are believed work here. So So there again, Dr. Harris in pharmacology will talk about the bisphosphonates coming up a little bit later. There it is, vitamin D. You're likely to see a question about vitamin D on board exams because of the integration with physio, biochemistry, and anatomy, and where things are happening, as well as the pathology of osteoporosis and osteomalacia and rickets. Major vitamin for boards. Now the next vitamin to talk about is vitamin D. Now vitamin D, I'm not going to go into too much detail here because hopefully it was discussed in neurophysiology, but the idea is there are 3 kinds of vitamin D. Now no one expects to know the structure of vitamin D, but what vitamin D has, it has a single ring and 1 long appendage. And on this 1 long appendage determines the name of the vitamin D and the function of the vitamin D. If on carbon 1 of that long appendage there is a hydroxy group or a carboxy group, it's then called retinol or retinoic acid, and these have nothing to do with vision. Retinol, retinoic acid, vitamin A's have everything to do with increasing cell division here. These are hormones that bind to receptors that cause the cells to grow. Now, see this blue arrow here, this blue arrow? You will experience this in your lifetime. You will experience this someday when you become an old timer like me. What I mean by this, someday in your life, you're going to get out of bed. And then when you get out of bed, you're going to go to your bathroom, look in the mirror. In the mirror, there's going to be these things called wrinkles. And when you see these wrinkles on your face, you're going to start to panic. Oh no, Father Time is coming in my direction. So you're going to get all nervous and all this kind of stuff. And you're going to get really depressed. And so what are you going to do? You're going to get on the phone and call up Mary Kay and say, hey, Mary Kay, I need some of those creams that you sell. So sure enough, Mary Kay will come to your house in a nice pink Cadillac. And then Mary Kay will come to your house in a nice pink Cadillac and sell you all these creams. and then she'll leave. So then after, so what happens is you buy all these creams and Mary sells, and you take these creams and you put them on your face where the wrinkles are. So you sit them, you put the creams, put them on your face where the wrinkles are, and then you let the creams sit there for a while, and then you wash away the cream. Then you go to bed that night, and then you wake up in the morning, look in the mirror, and all those creams have disappeared. How do you explain that? Well, we explain that by saying that when you take those creams and put them on your face where those wrinkles are, that my wife spends a fortune on. A fortune on. You know, I don't understand why she doesn't go to the store and go to Walmart and buy oil of Olay or something. She buys all those Mary Kay products. You know, I keep telling her, why do I mean, they're expensive. And she says, yeah, but they work. And I said, but, you know, but they're expensive. I mean, why do you think Mary Kay comes to the house with a pink Cadillac? She doesn't come with a Chevy. She comes with a pink Cadillac. And it's because they're expensive stuff. Buy that oil of Olay from Walmart. But anyway, the biochemistry is that you put that creams on the face, and the active ingredient is vitamin A, either retinol or retinoic acid, and they come into the skin, and the skin are cells in epithelial cells in G1 phase, and those retinoic acid will come into the cytoplasm of those epithelial cells, bind to the vitamin A receptor, and then go into, which is a zinc finger receptor and a specific transcription factor, and they'll go into the nucleus, turn on gene expression for epithelial cells. Epithelial cells grow and grow and grow, and wrinkles will disappear. There it is. There's the biochemistry of this blue arrow that you should be able to verbalize that answer. It's already been, that's right. Cindy Crawford stuff. That's right. That's amazing. All right, now what's coming now? So that's a retinol. That, you know how they work. These are hormones. Now, the other vitamin A is retinal. Carbon 1 is an aldehyde group, and that's involved in vision. Now, in neurophysiology, remember in the neuroanatomy as well, in the rod cells, remember those elongated cells in the outer membrane, there is that's where vitamin A is needed, because specifically it's called 11-cis retinal. That's the stuff involved in vision. And it binds to a protein called opsin to make the visual pigment called rhodopsin. And rhodopsin is in the stacked membranes of the rod cell. And rhodopsin, sometimes called the visual pigment, but sometimes called the photoreceptor. And the idea is when I'm sitting right here, and in my eye are coming trillions and trillions of photons in my eye. Remember, a photon is an electron. And where are these photons going? Where are these electrons going when they bombard my eye? They're colliding. They're going into the rhodopsin. And when they collide with the vitamin A in rhodopsin, that's where the electron goes. And then in neurophysiology, you talk about how the signal is then given to the optic nerve, and then you perceive an image. And then you get the color with the cone cells. It's the same principle, but that's all neurophysiology. But the biochemistry initial part is, where did the photons go? They went into the retinal, 11-cis retinal, part of rhodopsin. They collided with the vitamin A, and they set off a cascade, a signal transduction to the optic nerve, ultimately to the brain, to give the image that you are seeing. And when you don't have vitamin A around, and vitamin A deficiency, no retinal, what's the first symptom? Everybody knows this. What's the symptom of vitamin A deficiency? What's the first symptom of that? That's right, night blindness, the inability to see in dim light. You drive your car at night, you're not gonna, if you have vitamin A deficiency, what little photons are around, there's no vitamin A to pick up those protons, and that's the night blindness. That is the first symptom, ultimately giving way to blindness. No vitamin A, the photons have nowhere to go in your eyes. They bombard the eyes, but they can't be picked up by vitamin A in vitamin A deficiency. Therefore, ultimately, blindness will occur. On the other end of the spectrum, vitamin A is notoriously toxic, especially in pregnancy, in the developing fetus. Now, it's known that vitamin A has teratogenic effects in a developing fetus. And why is that? Because vitamin A in something like 2 times the recommended daily allowance, has teratogenic effects. And for a long time we didn't know, but now we know. And the reason is because it interferes with the homeobox genes. Remember the stuff we talked about the other day in molecular embryology? These homeobox gene encodes the homeodomain proteins that encodes that binds to enhancers of genes involved in placing limbs and so on. And vitamin A interferes with that. It has teratogenic effects. 2 times the recommended daily allowance. In other words, vitamin A is behaving a lot like thalidomide. It has these teratogenic effects here. That's also in adults or teenagers or whatever. Teenagers having a severe acne, and they're being treated with those isotretinoids, those very powerful vitamin A derivatives. You got to be careful with them because 2 of the 3 vitamin A's are hormones, and they can interfere with normal cell growth and differentiation. So you've got to be careful with these vitamin A's in pregnancy as well as in the treatment of acne, because they can, in excess amounts, can be very damaging to the cells in G1 phase here. All right, now, so that's vitamin A. Now, the next one I want to talk about in a little bit more detail is vitamin K. Okay, now vitamin K, has everything to do with vision, and you're going to hear about it next week with Dr. Barone in hematology. So now here is the biochemistry point of view here. Okay, now on this figure is the basics of vitamin K biochemistry here. Now it turns out in the human body there is only one cell type that needs vitamin K. There's an enzyme in that cell that needs vitamin K in order to work. And the only cell that needs vitamin K is the liver cell, because the liver has a major enzyme. Get ready with your highlighter. This enzyme is called the gamma-glutamyl carboxylase that needs vitamin K in order to work. And now what does this enzyme do? What happens is as you should know from hematology, there are 4 blood clotting factors, not all of them, 4 of them, including prothrombin shown here, as well as 2 other blood clotting factors, blood clotting proteins called protein S and protein C, and 4 blood clotting factors total. So the 6 proteins that must see this gamma-glutamyl carboxylase while the protein is being made on the ribosome. So in this example of prothrombin, this represents the messenger RNA for prothrombin. Here is the ribosome, and that purple line represents prothrombin protein being made starting with the N-terminus. Now remember, the N-terminus will have the N-terminal hydrophobic signal sequence that'll get that protein into the lumen of the ER of the hepatocyte, and while it's being made, so this is co-translational modification. While it's being made, this gamma-glutamyl carboxylase enzyme uses vitamin K. As the name of the enzyme says, it puts a carboxyl group on the gamma carbon of glutamate. All right, now again, this is being done co-translational while the proteins are being made. Let me ask you this: what's another example of the blood clotting what's another example of a protein, including prothrombin? Excuse me, let me back off. What's another example of co-translational modification of a protein? Let me see your answer here. Besides gamma carboxylation, what's another example of a co-translational modification of a protein? Well done, Takir, Hayal, and you're well done. That's right. Everybody else, that's right. It is N-glycosylation. So so prothrombin will be N-glycosylated. That's right. And it will also undergo gamma carboxylation on the glutamates near the N-terminus of prothrombin. So remember, here is the N-terminus. Here is glutamate near the N-terminus. And this enzyme is going to put a carbox group on the gamma carbon. All right, now I'm going to show you some structure here. Don't go panicking, but there's a reason for this. This is glutamate. Now remember, glutamate has 5 carbons. Here is the alpha carboxyl group attached to the alpha carbon. Here is the alpha amino group attached to the alpha carbon. Now the alpha carboxyl group, alpha amino group are in peptide linkage to other amino acids. Nothing strange. Now if this is the alpha carbon and it says the enzyme puts a carboxyl group on the gamma carbon, where is the gamma carbon? Let's count them out. Here's the alpha carbon, beta carbon, gamma carbon. So this enzyme is going to put a carboxyl group on the gamma carbon. You're going to say, hey, it already has a carboxyl group on the gamma carbon. Yes, but this enzyme puts on another one. And there it is as the product. Gamma, look at the name, gamma carboxyglutamate as the product. Gamma, that's the gamma carbon, gamma carboxyglutamate as the product. So by putting an extra carboxyl group on, and then the prothrombin is finished being made, here's the extra carboxyl group on near the N-terminus of the glutamate, and then prothrombin is then being secreted into eventually goes from the ER to the Golgi and out the body, out the liver cell. It goes into the bloodstream. All right. Now, so prothrombin has the extra carboxyl group on the glutamates near the N-terminus. Now the question is, why? Do you think prothrombin has an extra carboxyl group on it? So the prothrombin can say to all the proteins in the bloodstream, hey, albumin over there, hey, look at me, man, look at me. I have an extra carboxyl group on me. I'm nice and pretty, and you're not. Or do you think it's doing it for a purpose? That's right, it's doing it for a purpose. Now, what is that purpose? Now, what this purpose is all about is this harkens back to hematology reviews of the coagulation cascades, the extrinsic and intrinsic coagulation cascades. Now, if you were to go back to the extrinsic and intrinsic coagulation cascades, what you would find is, besides the blood clotting factor proteins that are needed for blood clotting, you need 2 other items that are necessary for proper blood clotting besides the proteins. And these other 2 things, medical students tend to forget them. But please don't forget them, because they both involve the gamma carboxyl group, and both of them are necessary for proper blood clotting. Now, the way you would figure this out is, what are the other 2 items necessary for proper blood clotting? The way you would figure this out is, you would say, by putting an extra carboxyl group onto the glutamate, you would think, well, you have extra negative charge. This must have something to do with making, with attracting something that's positively charged, right? And sure enough, if you go back to hematology reviews, you will find in very fine print, you will always see calcium, 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 calcium. Calcium is necessary for proper blood clotting. And there it is. It binds calcium. Okay, so then you got to ask the why question again. Well, what do you need calcium for? And what's the other item necessary for proper blood clotting? Now, the other item and why you need calcium is coming up in just a minute here. I'll get to that issue in just a minute in hematology. All right, now before we get to that answer is, now what about vitamin K deficiency? Again, you need vitamin K to put the carboxyl group on glutamate to contract calcium, as well as the other item necessary for proper blood clotting. Okay, but now in the absence of vitamin K, in vitamin K deficiency, the blood clotting factors are still being made. They still get secreted into the bloodstream, but they are useless. If you don't have this carboxyl group on the glutamate, because you have no vitamin K, it cannot attract calcium and therefore increase coagulation time. So they're still being made, but they are useless and therefore increase coagulation time. Okay, now to understand what is the other item necessary for proper blood clotting, to understand that, I got a question for you. And that is, if I took blood out of me right now, I took blood out of me and spilled it on this ground that I'm sitting down next to on the ground and come back next day to lecture tomorrow, what would that blood look like? I take blood out of me, spill it onto the ground, come back the next day. What would happen to all that blood? What would happen to all that blood? That's right. It would clot. All that blood would clot. It would coagulate, clot. Okay, how do you explain that? How do you explain that? That same blood in me is not clotting. I take it out of me and spill it onto the ground. It clots. How do you explain that? To explain it, you have to invoke the other item necessary for proper blood clotting. Now, this is not in your book. What I'm going to do is integrate biochemistry with some hematology here and explain why blood clotting here. Now, to explain this, I'm going to go to the whiteboard and explain this, not come back to this, come back. But here is the whiteboard. Now, what happens is, to explain the other item necessary for proper blood clotting, here is any cell in the human body. Let's say this is a red blood cell, the plasma membrane of any cell in the body. Now, remember from basic biochemistry, the plasma membrane is asymmetric. It's made out of an outer leaflet and an inner leaflet, but it's an asymmetric membrane, plasma membrane, meaning that the phospholipids on the inside of the membrane are the negatively charged phospholipids, whereas the phospholipids on the outer leaflet are the neutral phospholipids on the outside. What I mean by this is the phospholipids on the interior are the negatively charged phospholipids, like phosphatidylinositol, PI, phosphatidylglycerol, PG, phosphatidylserine, PS. The negatively charged phospholipids on the interior, PI, PG, and PS. On the outer leaflet are the neutral phospholipids, like lecithin, phosphatidylcholine, phosphatidylethanolamine. So the membranes are asymmetric. Now, if you think, well, so what? Stay tuned. It's coming up. All right. Now, also in your bloodstream right now, besides these red blood cells carrying oxygen around, we'll talk about beginning tomorrow, but also in the bloodstream is prothrombin. Suppose this represents prothrombin from the N terminus to the C terminus. So here's the prothrombin from here all the way to there. And near the N terminus is the gamma carboxyglutamate. You know how it came? That was put on, the carboxyl group was put on by vitamin K action. Now, what's going to happen is when you turn on the extrinsic-intrinsic coagulation cascades, what happens is they ultimately result in the conversion of prothrombin to thrombin. Thrombin, now suppose, is from here to here. Prothrombin has to be cleaved to generate thrombin, and then thrombin then goes and does its thing. Okay, but now the idea, so just picture right now, in your bloodstream right now are red blood cells carrying oxygen and prothrombin. They're not seeing each other. But when I spill blood out of my body onto the ground, all I need is for one cell to die. Cells will die, but all I need is for one of them to die. And when a cell dies, it lyses, it opens up. And when it opens up, the negatively charged phospholipids are exposed. Now they're negatively charged, and so is the prothrombin negatively charged. But now, what do you think bridges this? And the answer is calcium. The calcium already in the blood now comes and bridges the prothrombin with the membrane and holding the prothrombin onto the membrane. And if you go back to your hematology reviews, the other item necessary for proper blood clotting, it's membranes. You're going to see in very fine print, membranes, or you might see the words phospholipids, or you might see the letters PL for phospholipids. So you need membranes for proper blood clotting. Why? Because membranes are acting like a platform to hold the prothrombin on the membrane. And then the extrinsic, intrinsic coagulation cascades kick in. They then start to convert, all the cascades convert factor 10 to factor 10A, factor 5 to factor 5A, and these are proteolytic enzymes. And so all these proteolytic enzymes are now in very, very close proximity. They're all being held near the membrane, and they see each other. And when they see each other, they can cleave. They cleave each other very efficiently. Remember, PT is within 10 seconds. They start cleaving each other, ultimately generating prothrombin, the thrombin, then thrombin is then released, which then cleaves fibrinogen to fibrin, causing the clotting that goes on. But the concept is that all these proteins are in very close proximity. You can envision this like a matrix. A matrix of proteins are now held together in the membrane by ionic bonds. That's why you need the gamma carboxyl group by vitamin K, because you need to attract the calcium, and that's why you need membranes. Just picture, if you don't have vitamin K, there's not enough negative character of prothrombin, calcium won't bind to it, will not bind it to the membrane. And so therefore all these proteins have a hard time seeing each other, because prothrombin's way over there, factor 5 is way over here, factor 10A is over here. They have a hard time seeing each other, increased coagulation time. That's why you need the gamma carboxyl group. It makes sense, this hematology business. Why does blood clot? There it is right there, as simple as I can state. This is why blood clots when I take it out of me, because one cell dies, it opens up, and there it is. Okay, because when you take blood out of yourself, all you need is for one cell to die, it opens up, and then the negatively charged phospholipids on the interior are exposed. The calcium in the blood's already there. It then binds the prothrombin on the membrane by ionic bonds. And then the extrinsic, intrinsic coagulation cascades are kicked in, factor 5 and factor 10A, then sit in the membrane. They are proteolytic enzymes, which then cleave prothrombin to thrombin. Thrombin is then released. There it is. There it is, right there, as the blood is spilled. Now think about it. Think about it. It will make sense. Make sense out of it. You can't stare at this. You can't stare at this. You have to think about it. All right, now one more figure, then we're done. I'm going to go about 5 minutes over or whatever. About one more figure. I'm going to finish off with vitamin K. Okay, now what's happening here is, now by the way, keep in mind this figure, because suppose in a test tube I don't want blood to clot. It has everything to do with this figure. In a test tube, I want to donate blood for transfusion or whatever, and I don't want the blood to clot. Everything to do with this figure. Now on this last figure, major integration, extremely high yield figure, because we're talking about comparing vitamin K deficiency with vitamin C deficiency, major integration in hematology here. Now before we talk about this figure, as simple as I can state, what's the big difference between the mode of action of vitamin K and the mode of action of vitamin C? And here it is, as simple as I can state. Vitamin K is involved in carboxylation of some blood clotting factors, not all of them, some of them, whereas vitamin C is involved in hydroxylation of collagen. Talked about that last week. All right, now keep that in mind. Everything to do with the pathologies and hematology issues. Now in vitamin K deficiency, vitamin C deficiency, easy bruising and bleeding. But now with vitamin K deficiency, they have normal bleeding time, and vitamin C deficiency, they have increased bleeding time. How do you explain that in hematology? How do you explain that in hematology? In other words, if I were to cut myself and I bleed and I bleed and bleed and bleed, eventually blood will stop flowing at the site of the cut. So now the question is, what caused the blood to stop flowing? Let me see your answers here. When I cut myself, blood flows but eventually stops flowing. What caused the blood to stop flowing at the site of the cut? From hematology, what was the answer? That's right, it was platelets, not fiber or anything. It was the action of platelets caused the blood to stop flowing. Now let me ask you this. What attracts the platelets to the site of the cut? I cut myself and platelets go there. What attracts the platelets? That's right, it's exposed collagen. When I cut myself, collagen is exposed, and that's what attracts the platelets. And then the plates bind by the von Willebrand's factors and all these other things, and then eventually the plates then secrete things to cause the blood to stop flowing, talked about in hematology. But now, vitamin C, why do you have increased bleeding time? Why is it in vitamin C, blood continues to flow and flow and flow? Because you have defective collagen. Yeah, because initial hemostasis is mediated by plates, not the coagulation cascades. So what caused blood to stop flowing? It was exposed collagen, plates bind. But now in vitamin C deficiency, are you making collagen properly? Nope. You have defective collagen in vitamin C deficiency. Because vitamin C, remember we talked about it earlier. Why is it in vitamin C you have elastic skin? Because you cannot make hydroxyproline, hydroxylysine, then you can't do the cross-linking. And as a result, you end up with defective collagen. And if you have defective collagen, platelets cannot bind. And there it is. There it is. There is the explanation. It makes sense. Now, vitamin K has nothing to do with platelets. Nothing to do with platelets or collagen. That's why normal bleeding time. Because vitamin K has nothing to do with that. Now, another item here, it says increased PT. Now, PT is prothrombin time. Everybody knows that. The prothrombin time. Now, remember from hematology, what does PT measure? And the answer is it measures the extrinsic coagulation cascade. The extrinsic coagulation cascade. Remember, PT is a laboratory assay. I take blood out of me, put it in a test tube, and then add thromboplastin, also called tissue factor, that now turns on the PT, and blood should clot in about 10 to 12 seconds. 10 or 11 seconds, blood should clot normally. But in vitamin K deficiency, you have increased PT. Why? Because in the extrinsic coagulation cascade, you need vitamin K to make prothrombin and to make several of the other blood clotting factors. No vitamin K, no prothrombin being made properly, increased PT. But notice here with vitamin C deficiency, they have normal PT. Why do we have normal PT with vitamin C deficiency? Because vitamin C has nothing to do with the extrinsic coagulation cascade. Okay, is everybody with me? Everybody with me here? If you have questions, use the Q&A, but hopefully you're following me here. This should make sense. This is hematology issues, but the biochemistry explaining the hematology. PT is the extrinsic coagulation cascade, which depends on prothrombin. No vitamin K, no prothrombin, increased PT, which has nothing to do with vitamin C. There it is. All right, now another question for you. Here comes a question for you, if you're following me. What's the other assay, laboratory assay, that has to do with blood clotting besides PT? What's the other name of that other assay? That's right, and it's called PTT. Now, well done, everybody. Now, let me ask you this. What is PTT actually measuring? What's it measuring specifically? What's it measuring? That's right, the intrinsic coagulation cascade. The intrinsic coagulation cascade. Okay, but now why is the PTT not as useful when you're talking about vitamin K deficiency and vitamin C deficiency? Why not? Why not? Now, the way you answer this question is let me ask you this. What turns on the intrinsic coagulation cascade? What turns it on? What turns on the intrinsic coagulation cascade? Tissue down. I need to be specific, pure now. I need more detail. More detail. What exposure to what? What turns on the intrinsic coagulation cascade? And the answer is collagen. Exposed collagen. So when collagen is exposed, then factor, I think it's factor 10 binds, or 12. I think it's 12, I can't remember which one. But one of the factors binds with exposed collagen. And then it sets off the intrinsic coagulation cascade. So why PTT is not very useful? Because both vitamin K and vitamin C are involved in the intrinsic coagulation cascade. Vitamin C is needed to make collagen. If collagen is defective, intrinsic coagulation cascade won't work because collagen is defective. And vitamin K is needed for prothrombin. Prothrombin is needed by the intrinsic coagulation cascade. So if the PTT was abnormal, you would not know. Well, is it due to vitamin K or is it due to vitamin C deficiency? That's why it's not as useful there. Okay, now to finish this off, here in vitamin C deficiency, it has to do with fruits and vegetables. I'm not eating fruits and vegetables. Or the vitamin K. The main issue I want to get at here is breastfed newborns. Now, as you should know, breastfed newborns are prone to vitamin K deficiency and therefore prone to hemorrhaging. But now the issue is why? Why? And therefore, you as a pediatrician will be ready for vitamin K supplementation. Matter of fact, nowadays, vitamin K supplementation in a newborn is quite common here. But now we first want to understand the why. Why are newborns prone to vitamin K deficiency? And there are 3 reasons that contribute to vitamin K deficiency in newborns. And what's the reasons? There it is. I see a lot of it. Well done. Well done. There is some of the answers there, because the GI tract is sterile. The flora make vitamin K, but in newborn there is no bacterial flora. It's a sterile gut at birth, so it takes a number of days for the flora to kick in. Now, what's another contributing feature? I see it as well, because there's no vitamin K in breast milk. Daniel there, and other people, Miranda, that's right. If I miss you there, I apologize. No vitamin K in breast milk. They're not getting it that way. Another contributing feature is what? Let me see if anybody has an answer there. What's the third and last contributing feature? Why newborns are prone to hemorrhaging? Not in liver. Why not, Miranda? Why not in newborn's liver? Because it doesn't cross, that's right, being mature, but it doesn't cross the placental barrier. So the mother has plenty of vitamin K. The mother has about a month's supply of vitamin K, but unfortunately, in the liver, but unfortunately it doesn't cross the placental barrier. So at birth, the newborn's liver doesn't have any vitamin K because it didn't get it from the mother. So all 3 of these are reasons that contribute to why a newborn is prone to vitamin K deficiency and therefore prone to hemorrhaging. And therefore prophylactically, as a pediatrician, you're likely to give a newborn before they go home a shot of vitamin K just to make sure everything is okay. Now, just a couple minor things, then we're done. Not minor, actually, major, because the issue of farm and so on. Now, the idea here is now coming back to the normal situation. Now, we talked about a few minutes ago, if I take blood out of me and spill it onto the ground, it clots. Tonight, you should be able to verbalize the answer while it's fresh in your mind. You should be able to explain, without looking in the book, you should be able to explain why blood clots. All right, now suppose I don't want blood to clot in a test tube or in a bag that I might donate the blood for a blood transfusion. What causes the blood not to clot in a test tube? That's right. It's called in vitro inhibitors of blood clotting. Remember that picture I showed you, that diagram where the calcium is in the middle, bridging prothrombin and the membrane. And in a test tube will be anything that binds calcium, anything that's negatively charged, like heparin. Heparin, it activates antithrombin 3, but in a test tube, its main function, because there's a lot of sulfates in it, heparin sulfate binds calcium. Also sodium citrate, EDTA, these are substances that bind calcium, and by removing the calcium, those negative charges of the prothrombin repulse the negative charges of the membrane and increase coagulation time. It makes sense. And the very last thing is the pharmacology. These are known as in vitro inhibitors of blood clotting, but now here are the in vivo inhibitors of blood clotting, like warfarin, which is also called Coumadin, dicumarol, drugs like that. Now these drugs, how they work is a little bit more complicated than needed for your purpose. But all you need to know is there's a thing called the epoxide. Forget about that stuff. But the way they might do this on a national board is, what they might say to you is, this is a Lineweaver-Burke plot of a very important enzyme in blood clotting called the gamma glutamyl carboxylase in the black line. And in the red line is the gamma glutamyl carboxylase in the presence of warfarin. What type of drug is warfarin? And you say, well, you notice here that the lines cross, and what kind of drug is it? And it is a competitive inhibitor. Look how simple that was. So the idea is that warfarin looks like vitamin K. It competes with vitamin K action. That's all you need to know. Is don't worry about the epoxide issue. All you need to know is that vitamin K, that the warfarin looks like vitamin K. It competes with vitamin K action by acting as a competitive inhibitor of vitamin K action. And therefore vitamin K won't work. Therefore you don't get the gamma carboxyl group, increased coagulation time, and there it is. Blah, 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 blah. There it is. Get ready, get ready. It makes sense. There it is. Okay, everybody, we're done. That's why I was wondering why people put IMG, IMS. Now I understand. It makes sense. That's right. Okay, everybody, we're done for the day. So I'm going to, well done, everybody. Saw the high yield stuff. So tomorrow we'll come back and talk about vitamin E. And then we'll get in some big overviews on page 162 and 163. Major overviews, which will take about an hour or longer to go through those. And then after that, we're going to get into then we'll get into individual metabolic pathways after that. Okay, everybody, so we're done for the day. I'm going to post, I think, 4 questions on the Facebook when I'm done today. Post 4 questions on the Facebook. So you all have it was great fun being with you tonight. Thank you so much for attending, and hopefully it has been very beneficial for your purpose here. Okay, so I'm going to go, we're done for the day. Have a great evening and be safe, and God bless you all, and I'll see you tomorrow.